avec les travaux? Avec les travaux, ça va le son? Bon, je vais y aller. Il me regarde avec des gros yeux et des gouttes de sueur sur le front, mais je pense qu'il ne faut pas que j'en tire de conclusion. Cool. Vous me faites un signal en régie quand ça commence? 5, 4, 3, ok? S'il vous plaît. Bonjour. Ça, je pense que cette chaise, on peut même carrément l'enlever parce que tout à l'heure, il y a des modératrices qui vont arriver et elles risquent de se prendre le... Je la sors par derrière. Non, hein? D'accord. Vous enfermez. Caméra principale? Ou c'est celle-ci? Si je... D'accord. Donc si je dois m'adresser aux gens derrière leurs écrans, c'est plutôt celle-là. Good afternoon and welcome to this symposium, Chemical Exposome and Public Health. My name is Mathieu Rouault, I'm a French journalist and I will be your host today This event will be both in French and in English. This symposium is organized within the context of the French presidency of the Council of the European Union by the EHESP School of Public Health and the Université de Rennes 1 in partnership with the French National Institute of Health and Medical Research, INSERM, the French Agency for Food, Environmental and Occupational Health and Safety, ANSES, the University of Martinique and the European Digital University Alliance, and with the support of France Université. Bonjour à toutes et à tous, je suis très heureux de pouvoir, donc, comme je le disais, être avec vous pour parler d'un sujet extrêmement important, l'exposome. Vous le savez, une notion scientifique qui monte en puissance depuis plusieurs années et qui aujourd'hui prend la forme donc, de débats qui vont être nourris, intéressants à la fois sur la partie scientifique et institutionnelle. J'y reviendrai dans quelques instants. Développé dans les années 2000, le concept d'exposome a révolutionné l'approche et la méthodologie des scientifiques pour mesurer et surveiller les expositions humaines aux contaminants chimiques. Alors les intervenantes et intervenants cet après-midi vous le diront mieux que moi, mais je le dis en quelques mots tout de même, l'exposome est l'ensemble des expositions environnementales auxquelles l'homme est soumis tout au long de sa vie. L'exposome chimique est une partie de l'exposome. On en parlera plus en détail tout à l'heure. Un événement donc structuré en deux grands temps. Je le disais, d'abord, on va parler avec différents représentants des organismes qui sont avec nous cet après-midi, on les en remercie, avec eux de ce que ça signifie de travailler conjointement sur la notion d'exposome, de ce que ça signifie en termes d'articulation, de politique, d'échelons différents, local, national, européen. Et puis dans la deuxième partie, on aura la chance d'avoir des scientifiques de haut niveau qui vont pouvoir plus en détail nous expliquer un état des connaissances sur ces sujets. Avant de lancer à proprement parler donc, cet après-midi riche en contenu, je voudrais qu'on accueille ici même celui qui nous reçoit dans ce super lieu, qui est tout neuf. Il s'agit de Karl Bonafou-Murat, délégué permanent de France Université à Bruxelles. Bonjour et merci d'être avec nous. Un petit mot juste pour préciser l'endroit où on se trouve.
Bonjour à toutes et à tous. Et bienvenue dans la maison Irène et Frédéric Joliot-Curie. Nous sommes très heureux d'organiser aujourd'hui le premier événement que France Université tient, le premier événement dans toute l'histoire. Et j'ai le grand plaisir de vous accueillir et d'autant plus de plaisir que c'est un ensemble de partenaires qui travaillent ensemble. L'Université Rennes 1, l'École des hautes études en santé publique, l'INSERM, l'ANSES, l'Université de Masaryk, nos partenaires tchèques, EDUC, l'Université européenne. Bref, c'est un véritable écosystème qui est aujourd'hui présent à Bruxelles et je suis ravi de voir que ce qui se fait en Europe, sur le territoire français, sur le territoire tchèque, en Europe, est ici aujourd'hui présent à Bruxelles. Et puis dernière chose pour vous souhaiter la bienvenue, c'est aussi l'occasion de parler science en face à face et d'accueillir des grands chercheurs qui ont des responsabilités importantes. Le directeur de France Exposome, Michel Sanson, et la directrice de l'infrastructure européenne, Irene, qui est le professeur Jana Klanova. C'est aussi pour ça que nous sommes réunis cet après-midi. Merci et très bon après-midi.
Merci beaucoup pour cet accueil. Donc on est, vous l'avez compris, ici dans un lieu qui va compter de plus en plus ici à Bruxelles, donc la maison Irène et Frédéric Joliot-Curie. C'est une plateforme française de la science et l'innovation qui donc, va pouvoir pousser un certain nombre d'idées, dont des idées, des sujets en matière de santé et d'environnement. Je voudrais qu'on donne la parole pour commencer à recevoir ces représentants de ces différents organismes que je citais à l'instant. Laurent Chambaud. Monsieur Chambaud, bonjour. Venez me rejoindre, on va discuter quelques instants. On peut s'asseoir, regardez, hop, il y a des fauteuils, sinon on va nous demander ensuite pourquoi est-ce qu'on ne les a pas utilisés alors qu'ils sont si beaux. Merci d'être avec nous, vous êtes directeur, je le disais, de l'EHESP. Comment s'implique l'EHESP sur ces sujets maintenant depuis quelques années ?
Alors, on s'implique de façon très forte, notamment dans le cadre de ce qui se met en place en France et dans un cadre européen au niveau des infrastructures que l'on a développées à Rennes. Donc nous sommes partie intégrante de l'Institut de recherche que Michel Sanson va présenter. Dans ce cadre-là, pour nous, ce qui est important, c'est aussi de pouvoir alimenter en termes de recherche à travers notre plateforme technologique que l'on met à disposition de cet institut, mais aussi pour faire le lien avec tout ce qui tourne autour de tout ce qu'est la santé publique, c'est-à-dire à la fois de la formation, parce que parler d'Exposome, c'est aussi derrière travailler sur la formation et sur l'expertise. Et l'expertise qui est l'expertise dans tous ses sens, y compris l'expertise citoyenne.
Avec un mot-clé qui revient de plus en plus, enfin une expression en tout cas qui renvoie à un certain nombre de sujets de sciences très importants. Le terme est One Health. One Health, c'est un concept englobant dans lequel prennent part ces sujets sur l'Exposome. Vous regardez monter en puissance cette expression comment ?
Alors c'est un élément extrêmement fort pour nous, je dirais de deux manières. La première, c'est que l'Exposome, c'est une partie de ce que l'on peut voir dans quelque chose de plus large qui est la santé environnementale. Et l'école a cette priorité depuis qu'elle existe sur les aspects de santé environnement. Et donc dans ce cadre-là, c'est absolument fondamental pour nous que l'on alimente à travers l'Exposome une meilleure connaissance scientifique des liens entre santé et environnement. Ça, c'est pour ce qui est de la santé humaine. Mais quand on parle de One Health, on est d'emblée sur quelque chose de plus large, où on fait le lien entre la santé humaine, la santé publique humaine, la santé vétérinaire et la santé de notre environnement. Et c'est aussi dans ce cadre-là que nous, on souhaite s'inscrire fortement par des alliances qui sont des alliances en France, mais aussi des alliances européennes pour faire vivre le concept de One Health.
On est en direct avec, merci beaucoup Laurent Chambaud, on est en direct avec David Alice, c'est le président de l'Université de Rennes, bien sûr. Il est avec nous pour évoquer l'importance que recouvre à ses yeux cette notion d'Exposome, ces travaux menés avec l'ensemble des partenaires dont nous parlait il y a quelques instants Laurent Chambaud. Il est également donc l'Université Rennes est membre de l'Université européenne EDUC. Est-ce qu'on est en direct avec David Alice? Est-ce que c'est bien le cas? Bonjour Monsieur Alice, est-ce que vous m'entendez?
J'aurais aimé être avec vous physiquement cet après-midi, mais la venue du Premier ministre Jean Castex à Rennes avec le ministre Jean-Yves Le Drian nous a empêchés d'être présents physiquement. Mais nous sommes bien là et très heureux d'être là avec vous à Bruxelles.
Bon, ben merci de vous joindre à nous. Un événement important, hein, vous le disiez, que vous avez beaucoup poussé ensemble et qui se concrétise aujourd'hui ici dans ce lieu. Quel regard vous portez sur ces sujets? Pourquoi est-ce que finalement l'Université de Rennes se sent si concernée par cette notion d'Exposome en particulier?
Alors, si nous sommes tous là, je pense que c'est parce que l'Europe, elle a besoin d'une politique ambitieuse de santé publique. Et c'est la raison pour laquelle nous avons souhaité cet événement à Bruxelles pour sensibiliser les décideurs européens. Nos universités, organismes nationaux, grandes écoles, nous collaborons sur cette thématique au cœur de laquelle se trouve l'Exposome. Et si nous sommes là, c'est aussi pour témoigner peut-être d'une double ambition. Alors, une ambition rennaise, bretonne, mais au niveau national. Et là, je salue l'action en fait, de Laurent Chambaud, de l'EHESP, de Gilles Bloch de l'INSERM, de Roger Genet de l'ANSES. Ensemble, nous construisons aussi une grande université autour de la santé, en lien avec le numérique, l'environnement, autour de One Health. Et je tiens à saluer le rôle moteur aussi de l'IRSET. Et je voudrais associer d'ailleurs à Michel Sanson, hein, le nom d'un collègue qui s'appelait Bernard Gégou, qui nous a malheureusement prématurément quittés. Et je crois que Bernard aurait été très heureux d'être ici présent. Il aimait à citer, d'ailleurs sur faire comprendre le concept d'Exposome, il aimait à citer un peintre, Nicolas de Stel, pour dire « L'individu que je suis est fait de toutes les impressions, de toutes les expositions reçues du monde extérieur depuis et avant ma naissance. » Et on le voit, en fait, nous souhaitons mettre en avant ce concept d'exposome, et la crise sanitaire nous a ramenés aux fondamentaux et justement à la recherche, à l'innovation. Et puis, naturellement, nous avons cette ambition européenne partagée. Et là, je voudrais saluer les collègues chefs d'établissement de l'Université européenne EDUC, hein, de Masaryk en Tchéquie, de Potsdam en Allemagne, de Pécs en Hongrie, de Cagliari en Italie. Et je pense aussi en ce moment à nos collègues ukrainiens, à tous ces collègues européens qui sont mobilisés autour des valeurs de recherche, de science, de démocratie. Et je crois que notre université européenne, elle vise justement à constituer un socle, un univers de référence dans le domaine de la santé, en lien avec l'environnement et le numérique. Et je tenais tout particulièrement à saluer l'action de Masaryk, qui joue un rôle clé en la matière avec cette infrastructure. Donc pour porter cette ambition européenne, il nous faut des moyens. Et aujourd'hui, je crois que ce sont ces moyens autour des grandes infrastructures que nous portons ensemble. Et je le sais, l'ensemble des intervenants vont intervenir. Nous les avons un peu construits ensemble, et je pense à France Exposome et à toute l'action menée par l'INSERM, avec l'IRSET, avec l'ANSES, avec le soutien du ministère, mais aussi et surtout aussi avec EREN, l'infrastructure européenne que porte Masaryk. Donc aujourd'hui, c'est à la fois un aboutissement par rapport à cette feuille de route des infrastructures, à cette prise de conscience collective, et puis un point de départ pour faire avancer la recherche, pour augmenter notre impact, notre contribution face aux grands défis européens. Et pour nous tous, il est crucial que pour ces questions de santé, nous ayons des financements qui soutiennent des projets pluridisciplinaires pour réduire, prédire, promouvoir les impacts de l'exposome sur la santé humaine, ainsi que sur la santé globale, One Health, à l'interface des santé humaines, environnementale, animale. Et nous espérons vraiment que cette journée y contribuera. Et pour terminer aussi, je voulais évoquer aussi, en paraphrasant un peu d'ailleurs Bernard Gégou, l'exposome est souvent considéré comme intégrant des expositions négatives pour la santé. Et ensemble, nous avions aussi une vision que nous portons de considérer le volet positif de l'exposome. Et là, c'est là où nous sommes ensemble avec tous les organisateurs, la prévention, la promotion de la santé, qui sont à même de conduire à des exposomes favorables pour la santé, à l'échelle individuelle, du territoire, du pays et de l'Europe. Et en intégrant bien sûr tous les aspects socio-économiques. Je pense à Lao Tse qui écrivait « Prévenez le mal avant qu'il n'existe ». Eh bien, je crois qu'aussi notre conviction profonde, et c'est le sens aussi de cet événement pour l'Europe, pour cette Europe de la santé, c'est d'agir pour un exposome positif. Et je crois que la crise sanitaire avait montré que l'Europe avait peut-être baissé un peu la garde sur les financements de santé, et ça montrait l'importance justement de construire un avenir positif ensemble, et qui passe pour nous par cet exposome, par ces travaux de très haut niveau conduits par les organismes, conduits par les établissements au plus haut niveau dans cette perspective européenne. Et donc, je voudrais vraiment sincèrement remercier l'ensemble des directeurs, vice-directeurs, PDG, directeurs généraux ici présents, Laurent Chambaud, Gilles Bloch, Bretislav Danczak, Mathieu Schuller, et puis bien sûr Karl Bonafumura, qui nous accueille. Alors pour nous, c'est un honneur d'inaugurer cette maison Irène et Frédéric Joliot-Curie, et c'est une responsabilité. Et je crois que cette rencontre, elle en annonce d'autres pour une mobilisation collective.
Merci beaucoup David Alice pour ces mots. Merci mille fois, Monsieur le Président, d'avoir été avec nous, certes à distance, mais quand même bien avec nous. Et je voudrais justement, vous le citiez à l'instant, demander à Monsieur Danczak, Bratislav Danczak, de nous rejoindre. Vous êtes vice-recteur pour l'internationalisation de l'Université Masaryk, donc en Tchéquie. Thank you for being with us.
Thank you.
It's
Thank
a
you
great
for
pleasure.
inviting me, having me here.
So Masaryk University policy is driven by an 8-year strategic development document. It's an important document, and I would like you to explain to us what is it exactly, how this document, drive the exposome and the One Health concept, and how it takes into account this concept of One Health.
Yep. But Thank you very much for having me here in Brussels. I'm really sorry that I cannot continue with your beautiful language, but
I said both in English and French.
yeah, I'm trying to do my best. Masaryk University is preparing its strategic development plan for a certain period of time, and document must be approved by Ministry of Education, so it's significant and very formal document. And in these days, in document we prepared for period 21-28, the exposome and health environment, sustainability are absolutely core parts of this document, which means that we will devote, as we do, our research, educational activities, and also public outreach activities related towards city, region, and also whole Czech Republic to issues related to exposome or issues related to topic of our symposium. We are absolutely aware that Masaryk University should keep its leading position on in exposome for future. So Professor Klanova will talk about that later. We will even build up more infrastructural projects from National Recovery Plan for building another facilities for research and education, and I would like underline the educational activities which should follow directly from excellent research. So that's the formal, let's say, framework of our activities with exposome.
And what is Uni Biopharma app?
Biopharma Hub, it's something for what I'm in some way responsible as a vice rector. I'm not only for internationalization, but also statutory rector, vice rector of Professor Martin Bariš. Last year, we re-emerged Faculty of Pharmacy into Masaryk University. So we are now composed by 10 faculties located in different campuses, and crucial campus for this symposium is Bohunice campus, where is located Faculty of Sciences, where Resetox, responsible for exposome, is located also, together with Faculty of Medicine, Faculty of Sports, Central European Technological Institute, and new Faculty of Pharmacy. We hope that thanks to National Recovery Plan, investment roughly 95 million euros, we will build a facility for creating pharmaceutical hub for research, education, and applications. That's something what is absolutely crucial. We saw during COVID pandemic that we are weak as a university without technical background. We are very weak to answer quickly the questions emerge from pandemic, and from this perspective, after 4 years, we will hope to have at the end results for public, simply pills, or some tools,
yes,
which we can offer to our citizens.
And just a word about your collaboration with the University of Rennes 1. When did it begin?
Oh, many years ago. I was a very young student.
Okay.
when the cooperation started between two faculties with public administration, if I'm aware, maybe Sebastian will know better. Even there is a partnership between the city of Rennes and Brno city. In Brno there is a street called Renneska. So we built our partnership for a long time, and exposome is very good point on way of our cooperation for long term. So
because ecosystem like that take time to
and help to create another possibilities, activities. We'll meet again in May in Brno. So I think that you will see more and more results of our cooperation in near future.
Thank you very, very much. On est également avec Gilles Bloch, qui est donc président directeur général de l'INSERM, qu'on ne présente plus, en distanciel. Hein, il devait être avec nous et puis il n'a pas pu venir finalement. Mais je crois qu'on va pouvoir tout de même l'entendre. Bonjour, monsieur le président directeur général.
Bonjour, bonjour à toutes et à tous.
Merci beaucoup d'être avec nous pour parler de cette notion que vous connaissez bien, que vous portez à l'INSERM, celle d'exposome, une notion qui est pluridisciplinaire hein, dans l'âme.
Oui, absolument. Bon, l'exposome, ça, vous l'avez rappelé, c'est un ensemble d'expositions de nature différente, hein, dont l'exposition chimique, mais aussi les aspects sociaux, les aspects style de vie. Et puis, ça touche tous les organes, ça peut déclencher tout type de pathologie et puis ça peut même être bénéfique. Et donc, pour appréhender ce très large spectre de mécanismes et de manifestations, il faut bien sûr s'appuyer sur des outils, je dirais, classiques historiquement comme l'épidémiologie ou la toxicologie, bien sûr, beaucoup de biostatistiques pour établir des liens de causalité, mais aussi des disciplines auxquelles on est peut-être moins habitué à l'INSERM, hein, comme certaines disciplines de sciences humaines et sociales. Et tout ça, ça crée une palette multidisciplinaire extrêmement riche qui est très structurante pour un grand organisme national comme l'INSERM.
Et qui l'est d'autant plus que vous portez France Exposome. Un petit mot à ce sujet, on ne peut pas ne pas parler de France Exposome.
Alors, France Exposome, c'est un concept qu'on a construit ces dernières années avec notamment nos partenaires de Rennes et au premier rang desquels l'Université Rennes 1, le HESP, et je salue leurs responsables, et puis l'IRSET, hein, qui a déjà été mentionné comme une tête de pont de la construction de cette plateforme qui va permettre de caractériser l'exposome avec la puissance des outils analytiques modernes qui ont été localisés à l'IRSET, mais aussi dans d'autres laboratoires comme le l'IRES, le Laberca ou le Toxani.
Est-ce que vous diriez que ces travaux qui s'engagent sur l'exposome ont un caractère structurant dans vos collaborations avec vos partenaires locaux, nationaux, européens? Quelle place ça prend? Quel rôle ça joue?
Alors, c'est structurant à plusieurs niveaux. Bon, d'abord, il y a de nombreux projets européens, je n'ai pas le temps de tous les mentionner, mais qui ont tissé un réseau de partenariats extrêmement riche. Il y a des collaborations sur ce concept d'exposome qui se sont construites avec l'Université de Rennes 1, avec le HESP, on a mentionné tout à l'heure l'ANSES. Et donc, cette nécessité d'angles d'abord multiples crée la nécessité de coordonner des acteurs nombreux, sinon on n'arrivera pas à relever ce challenge. Et puis, au niveau national, ce sujet d'exposome, vous l'avez dit en introduction, qui est un concept qui se développe comme un outil puissant de découverte et de compréhension des pathologies, nous a amené à porter ces derniers mois la construction d'un programme national dans ces nouveaux programmes lancés par le gouvernement pour mobiliser toute la communauté française, donc un programme de recherche en santé publique ciblé autour de l'exposome qu'on appelle Expodis, qui va encore nous faire changer d'échelle en termes de structuration nationale et faciliter encore les partenariats qu'on pourra avoir à l'échelon européen. Et parmi nos acteurs nationaux, j'ai oublié de mentionner Santé publique France, hein, qui est également un partenaire très actif.
Merci beaucoup d'avoir été avec nous, Gilles Bloch. Je vous remercie mille fois et on va continuer bien sûr à évoquer les sujets que vous avez commencé à mettre en évidence au cours de cette intervention. Je voudrais qu'on donne pour terminer cette première séquence à Mathieu Schuller. C'est le directeur général du pôle Sciences pour l'expertise de l'ANSES. Bonjour, Monsieur Schuller. Je continue en français. On va parler avec vous du positionnement forcément un petit peu particulier de l'ANSES, hein, à la fois, j'allais dire, à la croisée des chemins entre les travaux de recherche d'un côté et puis les décideurs publics de l'autre. Un rôle pas forcément toujours facile à tenir peut-être.
Non, mais on le tient avec,
Challengeant, comme on dit.
on le tient avec rigueur et avec plaisir, parce que c'est effectivement notre mission que de faire la part des choses là où il y a de l'incertitude, scientifique, pour aider et éclairer le décideur public. Évidemment, aujourd'hui, on ne vient plus poser des questions ou demander l'avis de l'ANSES sur les dangers intrinsèques de l'alcool ou du tabac. Les ministères et les autorités de santé publique font ça toutes seules. Par contre, on a des questions là où il y a des incertitudes. Les incertitudes d'hier, c'était quel est le niveau de dose pour être un poison, lorsqu'on s'intéressait à une substance, le benzène, le monoxyde de carbone dans l'air, qu'importe. L'incertitude d'aujourd'hui, c'est les nouveaux dangers, les perturbateurs endocriniens, les substances cancérogènes. Quels sont les facteurs qui conduisent à l'accroissement d'un certain nombre de maladies neurodégénératives qui sont évidemment toujours très sensibles pour nos concitoyens. Et en fait, l'incertitude de demain, et c'est là que l'exposome a probablement sa place, c'est que les gens nous disent « mais oui, mais je ne suis pas exposé simplement à une substance, l'une après l'autre, mais à ce qu'on appelle l'effet cocktail », donc qui est peut-être le petit nom très commun par lequel on peut traduire exposome en face comme élément de réponse.
Ouais. Et alors, avec peut-être cette difficulté particulière en ce moment, j'allais dire, qui est que il reste, on retrouve ce terme d'exposome. Ça y est, dans la réglementation, ça commence à apparaître. Est-ce qu'il n'y a pas une étape à franchir supplémentaire en termes de pour instancier ce concept, le rendre plus concret, comment dire? Vous voyez ma question?
Oui, effectivement, on a un concept de l'exposome qui est particulièrement clair. Je pense qu'on aura l'occasion de le voir cet après-midi, de bien le partager. Il a été tellement clair qu'il est passé pratiquement directement de la paillasse des scientifiques au Code de la santé publique, donc dans la loi. Et donc, du coup, il s'impose à nous tous, aux organismes d'expertise, mais aussi aux gens qui sont des acteurs du risque. Et effectivement, pour essayer d'éclairer ça, l'agence a mobilisé son conseil scientifique et essayé de réfléchir sur finalement aujourd'hui, est-ce que comme M. Jourdain, on avait déjà de la prise en compte de l'exposome dans nos travaux, lorsqu'on fixe une valeur de référence sanitaire, on s'intéresse à la population la plus sensible, donc c'est déjà un bout d'exposome. Mais il y a encore des voies de progrès à faire, et le but de ce travail qu'on mène avec notre conseil scientifique, c'est aussi à la fois de faire progresser les méthodes, comment on peut identifier les substances qui, lorsqu'elles sont en mélange, font plus qu'une addition, ce qu'on appelle des substances synergiques, ou à contrario, celles qu'on peut subir les unes après les autres indifféremment. Ça, c'est une voie de challenge. Et l'autre voie, c'est aussi, il n'y a pas d'expertise aujourd'hui sans la recherche d'hier. Ou si je le dis autrement, il n'y a pas d'expertise de demain intégrant l'exposome sans que la recherche y contribue. Quelles données, quelle méthode, et ça, c'est un point très important aussi.
C'est ça les deux challenges qui vous attendent?
Absolument.
Dans les prochaines années. Merci beaucoup d'avoir été avec nous, messieurs, à la fois à distance et puis en présentiel. Je voudrais qu'on vous applaudisse pour vous remercier de votre présence, et on va passer, vous pouvez reprendre votre place dans la salle. On va passer, mesdames et messieurs, à un temps, in English please, de discussion avec Carmen Laplaza Santos, qui est chef d'unité santé, innovation, écosystème à la Direction générale de la recherche et de l'innovation de la Commission européenne. Bonjour madame, vous pouvez me rejoindre et vous asseoir où vous le souhaitez sur l'un de ces sièges. Et puis Carl Bonafumura nous rejoint pour discuter de ce sujet écosystème d'innovation politique européenne, déclinaison nationale et régionale. On va se demander comment mettre en musique ce sujet. Allez, je me mets ici. Mettez-vous là si vous voulez, ou là. Moi, je mets où on veut. Merci beaucoup. Thank you very much for this very interesting conversation that we will have right now. Broadly speaking, just first question to you, Carmen Laplaza Santos. Broadly speaking, what is the role of the DG Research and Innovation at the EU Commission?
Thanks a lot for the question. And first of all, merci beaucoup pour l'invitation. J'aimerais bien pouvoir m'exprimer complètement en français, mais je pense qu'il y aura certainement où je vais coincer. And that's why I will switch into English. So, well, I mean, that's a very difficult question to answer, or very easy in a way. It's very broad. So let me maybe just say that we are, of course, responsible for the research policy at European level, and that means responsible for funding a lot of research in order to ensure that Europe is building as much as possible on many different resources in order to ensure that we are really the actor in terms of research in the world. So we are responsible for the policy in terms of research, but we are also, and I think the most tangible way to see what our responsibilities are is our involvement in the European framework from research and innovation. Currently, the program Horizon Europe that we are responsible to put forward, to define, to design in terms of topic, but also to follow afterwards the implementation and the further exploitation of the results that come up. So we do that extensively with member states, obviously, how to define, how to put forward these programs, but also a lot with the other DGs of the European Commission. And maybe that links better to answer into your questions that, of course, funding research and supporting research is very important, but we also want to ensure that our research is contributing to the resolution of societal needs.
And how can we do that? How to control the fact that the research are...
So that's what we try to do more and more, engaging a lot with the policy DGs. So we are in constant dialogue with, for example, with our colleagues of DG Environment, our colleagues from DG Sante, our colleagues from the different DGs that at the end of the day will be able to exploit the results we are getting out of our research projects. And another thing that is very important for us is to engage also to the end users of those results. So more and more we put very much the focus into ensuring an intense dialogue, not only with the researchers, but also with ultimately afterwards they will facilitate the uptake of the research results for the benefit of the society in general at large.
Carbona Fumura, a question about the ecosystem. We have mentioned this word a lot of time just this afternoon. At the regional level, what does the ecosystem in Brittany represent as far as the Exposome is concerned?
At least two things. I'm not from Brittany, but I hope my Breton colleagues will confirm what I'm going to say. But first of all, there's a regional action plan on the relationship between health and environment, which was worked out a few years ago, and therefore this shows that there's support from the region and also from the region there's support in order to provide infrastructures, CPER, through the CPER, and also perhaps this very active support from Brittany as far as Europe is concerned. I mean, the region is very much involved in organizing the way higher education institutions and all the partners involving civil society, industries, political makers can contribute collectively to the European objectives of Brittany. So I think that's one of the specificities of Brittany, that is the high-level support to European action. Of course, there's support from all regions, but this is particularly visible,
yes,
I believe, in Brittany.
You've just mentioned infrastructure, and precisely this is my second question to you, Carmen Laplace Santos. In the future, do the study of Exposome will need new and specific research infrastructures, and how does the EU Commission help these infrastructures appear?
Well, of course, in the context of the Exposome, we are very active. Environmental health is a very, it's a field that we have been funding since more than 20 years. Close to 2 billion euros have been allocated by the European Union to a specific research on the impact of environment and health. And the Exposome has got an important part of that, in the sense that around 100 million have been, since 2020, allocated to 9 projects that are really looking closely to the Exposome. And I think representatives from those projects, those who are currently coordinating the cluster, will be here today afterwards. But we are doing many more of research in the context of environmental health, farther than the pure Exposome, and in many aspects, the research that we are funding would contribute really significantly to the Exposome needs in terms of research. So I think that farther than the creation of infrastructures, it's very important to link the diverse initiatives that are already ongoing in the field to ensure that all the aspects, that first of all, that we are not reinventing the wheel, and that for me, I mean, for us, collaboration, engaging as much as possible with different initiatives is essential. But nevertheless, we are really very, enthusiastic to see that there is a future research infrastructure in the making, in the preparation, IRENE, that we'll also talk later on. And we are really looking forward to being able to collaborate with them, because indeed having a research infrastructure would certainly facilitate this need to link between the different initiatives, to serve the needs of the different research groups on this field. And yes, we are very happy to see that they are in the roadmap of the ESFRI. One colleague of the Commission will come later on to talk about that. So really looking forward to see how the preparatory phase goes and how we, from our cluster 1 in health, from Horizon Europe, we can also engage our different activities with what IRENE will be doing.
Mr. Banafo-Murat, let's look a little bit more about another good example of what can be done at the at the national level, at the regional level. Let's talk once again about the University of Masaryk in Czechia. It's also a good example, as I said, of a university supported by its whole territory. And
again, I do not come from Masaryk.
No, you don't.
No, from Brittany, but the University of Masaryk and Brno, which is the second biggest city, I think, in Czech Republic, is very well known for its support of the university. And what is interesting is that the birth and the development of research on exposome in that specific area has something to do with the history of the city, because 30 years ago it was a city part of a mining territory, and of course the transformation of the mining industry has led to research on the impact of the mining industry on the environment, and therefore this has led in turn to the development of a research combining health and the environment. So this shows, just to show that it's very interesting to see that two ecosystems which have some strong specificities are connected through that kind of research. And secondly, it shows that the notion of ecosystem has something to do with history as well. And I'm sure the same could be said about Britain and why research on exposome came to develop in Brittany. I mean, the link between health and environment in Brittany is well known. So this historical depth, I think, is important in order to understand this concept of ecosystem. Of course, it's about contemporary research, state-of-the-art research, but it also has an historical dimension that should be taken into account.
Thank you. This is the end very soon of our roundtable. Sorry, the time is over, but one last question to each one of you. If you had one advice to give to scientists, industries, universities on the national level about the better approach to articulate and coordinate the actions to promote the research on exposome, what would be this advice? You have 2 hours.
No, for me it will be in the same line of what I said before, link with the very different and relevant initiatives for the field of the exposome. I can mention the different clusters we have. There is the exposome cluster that we'll present afterwards, but there are several clusters at European level ongoing now, looking at the impact, for example, of the microplastics and other sort of exposures. That is very important as well. Looking at the impact, in particular, urban environments, looking at the impact of endocrine disruptors. I think all these different initiatives need to be linked, and if I was a researcher now in whatever university in Europe, my advice would be engage with the responsible of those projects. And actually we can provide the information of the repositories of these different clusters that maybe some of you already know. Important to know as well that we are constantly putting forward new topics, and in particular last year the 3 topics that would be relevant would be the one on the understanding of the main exposures from air pollution, inside air pollution. We have electromagnetic fields, also research that is going going to start very soon. And of course, we have also an important co-fund, PARC, to assess the risk of chemicals that will start in May. I think someone will come afterwards to talk about this important project. So anyway, I mean, all in all, to say that there are many initiatives, and I think linking to them,
pull together,
it's what would be my advice. And of course, follow our actions in terms of support to research.
About collaboration, my last question is about EDUC. The University of Rennes 1 and the University of Masaryk are both members of the European Digital University. How can they share their expertise to the other members? Is it pertinent to do so?
It certainly is, and I think that one of the great assets of European universities, of which EDUC is a very good example, is that, well, they have to translate top-level research of the kind being led by Exposome into study programs. And this is a way of achieving this cohesion that you mentioned. You have to entrust and trust students in order to carry out, you know, future developments, and you have to teach them what are the new issues about this kind of research, so that you can train them, and by training them, you will expand the community of people being able to work on the research on Exposome, and this will have an impact on all the other member institutions of EDUC. So this is a kind of snowball effect that you have to get started, and this will certainly give results.
A huge thank to both of you, Carmen Laplaza Santos and Carl Bonafumura. Thank you very much for this very short conversation. It was a great pleasure. Let's continue this event. You can applaud them, please. Let's continue this event avec, in French, Monsieur Robert Barouki, qui va nous parler d'Exposome, bien sûr. The next step is Exposomics. That's it. Vous pouvez y aller, bonjour. Merci mille fois, franchement, Monsieur Barouki, d'être avec nous. On vous laisse vous installer. Et puis, je vais vous retrouver tout à l'heure en ce qui me concerne. S'ouvre maintenant donc la séquence à proprement parler scientifique. Je vous retrouve tout à l'heure pour la séquence de conclusion. C'est Madame Klanova de l'Université de Masaryk qui va pouvoir me remplacer en termes de modération. Merci beaucoup. Thank you for being with us. Et je vous laisse et je vous retrouve tout à l'heure. Et je vais écouter avec beaucoup d'attention et d'intérêt ces sujets. Merci beaucoup.
Use this one? Yes. So, good afternoon, everybody. Thank you for having us here. And let me introduce Robert Barouki, actually, who I am to introduce you, Robert Barouki, because all of you probably know him better than me. Robert is a biochemist, toxicologist, working both in INSERM and Dakar University, but he is also coordinator of many European projects and initiatives, including HERA project that was mapping the research needs in the area of environment and health, leading the pillar on linking environment and health in the HBM for Europe joint program under Horizon 2020, very active in negotiation of the PARC initiative in the last 2 years. So he is excellent scientist, but also he contributes a lot into translating the research results into the policy, linking the research and legislation in Europe. He is a good scientist, a good friend of mine. Floor is yours.
Thank you very much. After this introduction, I can, you made my day, Jana. Thank you very much. So, I mean, today I feel like I am from Masaryk, I am from Brittany, that's the feeling I get. And really thank you very much, particularly to Michel for the invitation. Okay, so the next step, the title, I didn't choose a title, it was chosen for me, which is nice, the next step in Exposomics. So let me go through, and if I'm too long, Jana, don't hesitate to just stop me. Well, I think it was mentioned in one of the introductory speeches that the environment is not only about bad things. The bad things are on the top of my slide, and the better things are on the bottom of the slide. It can be water, it can be green areas, social environment, etc. So let's not forget about good things. However, during all the rest of my talk, it will be about the bad things, and that's, and you know, pollution is not such a good thing, and we know, but it is important, and I think the link between pollution and exposures and environmental stressors and health is really extremely important. This is a slide from WHO, and it was not obvious maybe 10 years ago, I mean, that this would be the kind of slide that we would see, that pollution is actually one of the most, actually the most important quantitatively determinant of death worldwide before smoking and before the combination of a number of infectious diseases. So this means that we should really care about pollution. It's mostly actually in this, it's mostly air pollution, but it includes, you know, global pollution in general. So this is something we should care about and work on. There are many issues with pollution and environment, including political issues, and you know how political and strategic items are important these days, but one of the big things is that the countries that produce mostly the pollution are not the countries that suffer most from the pollution, and this is a big problem. So when you work on environment and exposure, you also work on politics, and that's really important. If we go now to the chemical aspects, the chemical universe, and this is a slide that was taken from the you know, chemical strategy for sustainability in Europe. And let me say here how much Europe has done in the world of environment, you know, and all these strategy, the zero pollution plan, the climate issues, I mean, how important these are. And one of them is the chemical strategy for sustainability. If we achieve that, it would be really great, but let's try to do it. In that strategy, there was a figure taken actually from the European Environmental Agency, I call it the iceberg, where you can see that out of, let's say, 100,000 chemicals that are currently on the market as a rough number, we know a lot, about 500 of them. Most of the researchers work on these actually. We know a lot about 500 out of 100,000. We know a little bit about 10,000 more, maybe a little bit more on 20,000, but for 70,000, we don't really know much. So there's even in maybe the most worked on stressor, the chemical stressor, even in that field, there's a lot to be learned still now. So this is really critical for the future. This is why, in a way, one of the reasons, and there are many others, Christopher White, who's an epidemiologist working at the IARC in Lyon, at that time, he was the director, he proposed this exposome concept. And he works in the field of cancer. And in the field of cancers, they knew quite a bit about the genes, the genetics, the genotoxic compounds, etc. I mean, there was a lot known about these compounds. But then, when you take all the other determinants of cancer that are not genetic, it was a sort of a mess, let's say. I mean, there were, you know, chemicals, there were physical stressors, infectious, diet, all kinds of stuff, and it was a mix and it was not clear. So he thought that if we put all these together, including some social stressors, maybe this concept will push people to work more on it and give it maybe some more substance. So he defined the exposome as all life course environmental exposures from prenatal period onwards, all life course environmental exposure, which includes internal body processes, external exposure, lifestyle factors. So it includes chemicals, physical, biological stressors, but also psychosocial stressors. It's a huge concept. And it includes the time dimension, all exposure over our whole life. That's very important too. He published his paper. He couldn't publish it. Actually, when you ask him, he couldn't publish it. Even nobody wanted, what is this? I mean, the exposome. And he ended up publishing in a paper where he was the editor. It's easier when you are the editor. And for 5 years, he told me that nothing much happened. Nobody cited that thing. And then suddenly Europe, the United States, people said, well, that may be a good idea. Let's try to see whether we can fund some projects on this. And that's then how it started. But when in science, when you give a sort of big concept like that, what's really nice is that you have all your other colleagues who say, I want to add something to it, or I want to make it a little bit more concrete, which is nice. And this is what the colleagues did. And you have this analytical vision of Rappaport and Smith, where they focused on what they call the internal chemical exposome. Trying to know as much as we can about the internal exposome would tell us a lot about all the other exposures. That's at least practical. You can do that. Miller was a toxicologist. He is still a toxicologist, Gary. He's a good friend. And he, as a toxicologist, he insisted not only on, well, all these cumulative exposures, and particularly the chemical ones, but also on the biological responses to those exposures. So then suddenly the exposome was a little bit more than about the exposure, but on their impact. I will get back to this a little bit later. And then you have the ecotoxicological vision, people working in the ecosystems. And here you have something called the eco-exposome. We mentioned One Health a little bit earlier. It's pretty much the same. It's about the interaction between the human and the ecosystem, both ways interactions, and how you have this vision of the exposome, both environmental vision as well as the human vision. And later, more later, I mean, there are a number of papers that I'm citing here, two of them, like from Elliott Price et al. And Vermeulen et al., pretty much the same, putting the exposomics in the larger field of the omics, metabolomics, transcriptomics, epigenomics, etc., putting it in the same field, working on network science, and also distinguishing, which is important, something called exposomics from something called functional exposomics. Exactly like in the people in the genome world, they distinguish between genomics, structure of the genes, etc., and functional genomics, what these genes are doing. This here, we distinguish about the exposome, I mean, all the exposures, but also what is their biological and health effects. That would be the functional exposomics. So nice. And I'm citing a paper that written by Paulo Vines. I have contributed, we worked on it together. I didn't imagine in my life that, writing a paper citing Bourdieu, who is a French sociologist, and because he, but that's because Paulo is so much involved in philosophy and sociology too, that, Bourdieu talked about the capital, the social capital, and here we talk about the, environmental capital all over our life. So these things add to the exposome and make it a little bit more concrete in certain way. So the history of the exposome is shown here. You can see 2005, Chris Wilde suggested it. We are a little bit over the puberty now. The exposome concept is 17. We are on the road to maturity, hopefully. And for 5 years, nothing. And then suddenly something's happening in the United States, other things in Europe. And I'm citing them. I will not go through them together. But the most, the latest things are, for example, the cluster, the EHEN cluster, the exposome cluster in Europe, extremely important. And Martine will talk about it. IRENE, that was mentioned a number of times. PARC also was mentioned. I just added, there's a journal now called Exposome. I mean, it's fantastic. This means that we are now in maturity is there. It is a complex concept. We have to talk about the stressors, where they're coming from, their source, where do you go? It's different if it's at home, at school, in the metro. Which time? Extremely important in toxicology. These are real important concepts in toxicity and toxicology. The routes, the distribution, the targets. So it is a complex. So how to study it? I've taken it from Denis Saragianis, this slide here, just describing the different tools that we are, having now, either for getting satellite images on the weather, on light, etc., personal sensors, environmental sensors, of course, exposure modeling to a large extent, the whole bunch of things that we can do now. I mean, in omics technologies and in kinetics that we can do here. The epidemiological work, and much of the exposome, we have to acknowledge, has been started by epidemiologists and made a large progress due to epidemiologists, and Martine will tell us more about that, but also the interaction between epidemiology and toxicology to give more strength to causality. Exposures are so complex. There are a lot of biases there. You need to strengthen the causality between an exposure and an effect. So these are kind of approaches that are done. So they said, well, the next steps, I think we have to build a very integrated exposome program, ideally internationally, but at least at the European level, because Europe has such a big deal, and at the Brittany level, of course, certainly. But I think we have to do that. And I'm suggesting a number of steps. Nothing surprising there, but at least let's try to. First, the infrastructures and toolboxes. I'll get to that later, and other speakers will too. Then I think we have to integrate in this exposome framework some of those extremely important global challenges that we have: climate change, biodiversity loss, the global pollution, etc. It's not always integrated. We have to do that. I mean, it's important. We have to develop some specific scientific objectives, like building a stronger alliance between exposure epidemiology and toxicology, linking to clinical medicine in many cases. I'm an MD, so it's important for me. I think it is important to link to the medical stuff. The time dimension is extremely difficult. Epigenotoxicity, I think it's the epigenome is really important. And this, what we call the social to biological transition, bringing social stressors in line with what we can do in terms of biology. And of course, there are a number of regulatory public health issues. Let me cite one, the exposome risk score. It was not from me, it's from the Roel Vermeulen and paper in Science. They suggested that, and I think it's worth to think about it a little bit. So, of course, infrastructures, yes, Irene will be an example. I will not dwell on it. I think Jana will describe it, but it is extremely important for us to do that. The EHEN, the exposome network, a number, you cited 9 different projects, more than 100 million euros, and also a toolbox. I mean, a number of development tools that are developed in this network, which will be extremely important for all of us, So I think we hope that this network will really succeed in developing all this. It's extremely ambitious. I think they have the means. They have a number of excellent teams in there working on this, and I hope this will be also extremely relevant for all the exposome work in Europe and worldwide. Now, if I focus on the chemical exposome, because there's much that has been done, and this is a picture actually from Gary Miller, comparing all the variables that we have in the genome field, that it's more than 1 million different differences between human, polymorphism between human, and comparing with the number of chemicals that we can assay in a run. We can do a few hundred, maybe a few thousand now. We are starting to get to the few thousand, and sometimes with some doubts on who is what, but still we can do that. But it's still far from the number of variables that we have in the gene field. And the ultimate hope from many chemists and analytics is to get as much as we can knowledge on these different chemicals and be in the tens of thousands of chemicals that we could do. So that's a tool that's important, and that people are really working on. I said toolboxes, I said infrastructure, but also including these major challenges that we have, climate crisis, into the exposome field. Because this is a picture from a very well-known, Andy Haynes, a very well-known paper on climate change and health. And if you look at the top line, it's only about exposures. I mean, it's simply about exposure. Air quality, heat stress, water quality, food, etc. I mean, infectious agents, etc. So it is really about the exposome, and we have to, put our framework of exposome, put the climate change and the climate crisis completely in it. This is on biodiversity, and basically it's the same thing. As you can see under the green box there, you have exposure and experience. Exposure is so important in biodiversity loss. There are differences in the exposure due to the biodiversity loss, and this also can be integrated in a larger exposome framework. I don't know if this means that I'm too long and that I have to let's see how it goes. Of course, the other really critical things in terms of global changes is the urban environments, and there are a lot of, for example, programs in the EU about urban environment, but also this is also essentially also about how the people living in cities, how much they are exposed to stressors, all kinds of stressors, chemical, physical, but also sociological stressors. This is extremely important too. Let me go a little bit more into detail about some of the some of the tools and some of the themes that should be developed within the exposome. This is something that I usually present when I speak about in HBM4EU or even PARC actually. The AAA, because aggregated exposure pathways, this is how we describe the exposures and how they aggregate and end up in an internal dose in human. all that the descriptors that describe the internal dose in toxicology, which we call ADME. And now, concerning the health effects, we talk all the time about adverse outcome pathways, AOPs. And I think all these should be put together. And I think both HBM4U, but also PARC, will help putting together the sources, the pathway leading from exposure to the internal dose, and then from internal dose all the way to the adverse outcome. This is, I think, would be a very nice aim for all these projects. Now, there are many mechanisms. This is also from a very good paper on the hallmarks of environment and health. There are many mechanisms that can lead to issues, and they are described there. I just added a few of them to your left. And I think these are the focus of our work, because as I said, the exposome is not only about exposure, it's about also the pathways that lead to disease, and we have to work on this also a little bit more. I will not describe because we don't have really time to go into detail. There are some major issues, of course, in the field of exposome and chemical exposome in particular. One of them is the issue of mixtures. There's a lot of work in Europe on mixture, and there's still much more. It's extremely important mechanistically, of course, but also at the regulatory basis. There is a lot of thought about this and in the regulation and in the regulatory field, including at ANSES and other, at EFSA and many other agencies. It may be extremely important because suddenly we will realize that some of these limits that we are providing for each compound, because we work on each compound alone these days, may be too low if we consider the mixture. And Andreas Kortenkamp describes this as saying something from nothing, which is, when you mix 3 or 4 or 5 chemicals, you can get an effect when you thought that each one alone would not give an effect, something from nothing. We have a different way of saying it in French, but I will not dwell on that. Time is an extremely, I think in the exposure field, the issue of time is extremely important. And it's really not so easy to model the effect of exposures with time. It's not so easy. There are many different ways where you can imagine how the fact that we need 10, 20, 30, and more years, sometimes generation, to have an effect out of an exposure. Either because you are exposed, you need to be exposed for a very long time to get an effect. That's, the reiteration of the exposure. Sometimes it's the persistent inside your body, case of dioxin, for example. But most importantly, I think, it's what we call the programming, the developmental alteration by some chemicals or other stressors, leading to something that does not appear immediately in terms of health, but an increased risk for a health effect many, many years later, sometimes maybe a generation. Exposure during the fetal period, very short in time, and then an effect much later in life for an increased risk of disease. And here, this is why called the developmental origin of health and disease. There are many people working on this, and it's not only about chemicals, other stressors, social stressor. Sometimes during the fetal period or during the when children are extremely young, it's really important that these stressors lead to a health effect sometimes much later in life. And epigenetics, this is why I mentioned epigenetics in my talk initially. Epigenetic is probably the likely mechanism, and we should work much more on that in the future. I mentioned the fact that we have to link some of these social effects, psychological or social effects, to biological pathway. I'm giving you here just an example. It's a process that is described called social defeat, where you can mix aggressive animals with nice animals together, and you have the defeat of the nice guy because of the aggressive guy. But this can be translated in terms of biology. I mean, here, for example, it's brain-derived neurotrophic factor that is decreased in the social defeated animal. And BDNF is also a target for chemicals, and we have worked on that in HBM4EU. So the same biological pathways are targeted both by some psychological or social stress, but also by chemical stress, and we have to work on the combination of these different stressors. So there are extremely important implications in regulatory science. At ANSES, there is a group, as was mentioned earlier by Mathieu, there was a group working on how can we translate the exposome concept for a regulatory agency. That's a major problem. And there are many implications. Of course, mixture is a big issue. I think that's where the application will start. But the fact that there are multiple stressors that can interact, I mentioned the exposome risk score, something that we have to work on. There are the vulnerable populations. That's also extremely critical for agencies. Public health messages. How can we communicate to the public, these kind of complex concepts? And what are the implications? I mean, for example, the usual example is when we ask people to eat vegetables, which is extremely nice, and when the toxicologist comes and tells us that pesticides are not so good for your health, it's a problem. So we have to solve these problems. Precision prevention, we have now these detectors, these scanners that you can have on you, and that can tell you every time, how much you are exposed to particles or to those chemicals. How can we use that? It's extremely precise. Like they do precision medicine with genetics. Why can't we do that with exposure? And also we have some more globally ethical responsibility for future generation, because what we decide now will have an impact on the future generation. And I think it's really an important one. This is a description of the different, generation. We call them the baby boomers, the Generation X, the millennials, Generation Z. I think the next generation will be CC for climate crisis, because they will hear a lot about that. They will hear a lot. It will change their life. And if I had to choose one letter, it would be CC. I tried to call it Generation X, EX, like exposome, but I think it's probably climate crisis that will be the thing. I think ethically, we have this responsibility and we owe it to the future generation. And with that, I would like to thank you very much.
Thank you very much, Robert. I think we have time for some questions. So are there any questions from the audience? If not, I can ask the question in the meantime. Robert, if you now had a choice and you can ask the European Commission for one project that would be granted to you, what do you think is the most important thing, where we should start? Because what you introduce is the lifelong program for all European institute for another decade. So if you had to make just one choice.
That's a tough one, because it's really difficult to choose one thing, as you know. It's really complex. I think the easy thing is find a process to coordinate all what's happening. I mentioned EHEN, I mentioned IRENE, I mentioned PARC, and you're right when there are also the EDC programs, there are programs on social sciences, urban life, etc., and all these should be. Each one is working. So coordination is important, but coordination doesn't produce new science. It helps bringing together, some of the science. If I had one big project, I would launch a very big project on epigenetics. With all stressors, social, chemicals, etc. I think this will be, if we can develop epigenetic tests that will tell us more about what people have been exposed to and eventually what are the risks for the future, I would do that. But this is because you asked me to choose one. Voilà.
Listening to you, maybe we should launch one big project for education and PR to educate people how those chemical exposures are important. Because, people hear about the Green Deal, they hear about climate change, but very often they associate Green Deal with the climate change only. Oh. And they fail to see how many other elements there are, and chemical exposures are behind most of them. So I think this is something we should really work on together.
You are absolutely right, and I'm really happy you said it, and I didn't say it, but I'm really happy you mentioned that. It's very important.
Thank you very much for your presentation. It was very interesting. Just one question which came to me as I was listening to what you said about time. What could be the contribution of the social sciences and humanities to those projects? Because I think there are some of them which require the participation of such fields of research.
Well, I think almost all of them do. There are different ways where you can bring in, I think, social sciences. First, I mentioned a number of times social stressors. these are extremely important, especially in the beginning of life. I mean, also, but we do know that, for example, people who have limited, economic status or difficult economic status are usually have a higher risk to be exposed to other environmental stressors. This is one way where this works. But also, we know that the social stressors converge with other stressors to, biologically and even on health. So these are really important. But also, this is one aspect, but there are many, many other aspects. For example, it is extremely difficult to convince people that what they were exposed to 30 years ago has a very long, strong impact on their life, because it's too late it's too late in many cases. Yet, that's where it's really important to mention the good and the bad environment. We mentioned the bad environment all along, but there is a good environment. And the important thing about environment is that, in a way, these processes are, in a way, to a certain extent, reversible. You can do something about them. I mean, this is why it's so important, for example, in the determinants of cancer, to work on which are the environmental determinants of cancer, because you can do something about them. And that, I think we have to work on it a little bit more. I mean.
Thanks a lot, Robert, and it was really fascinating. Just a question, because you just mentioned something about the ethic issues. Do you take into account in your research the ethic issues, and what can we do just to put this forward, and even for the political decision, the ethic issues of such public health exposome issues?
Well, I hope I do, but I don't know. I'm not going to dwell on it. But what I meant here by ethic is the responsibility towards the future generation. This is exactly what I mean. I mean, we have an ethical responsibility toward the future. I mean, because all the decisions we are taking now will have a major impact. All the fact that we have made our climate much worse, we would not suffer that much from it. I mean, at our age, we will not suffer that much. But the next generations will suffer a lot. So we have a sort of debt to them, and therefore we have a sort of ethical imperatif, I would say it in French. I mean, ethical responsibility to make sure that we do not make things worse, and at least we start trying to make them a little bit better. This is what I meant, and I think it's really an ethical issue in terms of philosophy, but different maybe from the other ethical issues that we have in the labs. But
Thank you.
So, thank you very much, Robert, for your very nice and dynamic talk. I have a question, because you compare exposome, chemical exposome, with a genome project, and we know that it's just to describe the human genome, it's 30, 40 years. So to link the prevention to the chemical exposome, do you think it's a next step of the next 30 or 40 years?
Well, first, if you look at the history of the genome, it took much less time than people expected that it. I mean, to delineate the human genome at least, and then of course all the other consequences took more time. But I mean, I remember that, and there was this competition between Craig Venter and the Harvard, and it was. It took less than, it was less than 10 years, and at that time it was really at least. So I think it can take less time than we think it might. And you have to be extremely practical and extremely realistic. There are things that we can start doing now, that we know we can start. I mean, for mixtures, we know that we can start taking some decisions very quickly. Maybe it will not be the best decision. More research will be necessary to refine, to fine-tune the kind of decision we are taking. But I am convinced that we don't need 30 years to take decision. We can take decision now. It's as good as it is with the knowledge of today, but we have to push forward a big exposome program for the next future. And with that, we can do much better in 10 years and much better even in 20 years.
Also, thank you, Robert, for the very clear and dynamic presentation. My question would be on the exposomics you have shown. Exposome you have shown is the integration of the whole time history, but we are used to quantify detriments, hazards, risk, what do harm our health. But it has been already pointed out by different speakers this afternoon that there is also, if you want to be in the perspective of public health, to be able to recommend things that are positive for health.
Yeah.
There are not many works done for quantifying these aspects.
Well, it's starting. I mean, you know, if you look, it's amazing. I mean, whenever you go to the doctor, he tells you, you should do more physical exercise. I mean, whatever you have, you should do more physical exercise, which is true. As we know much more how better doing physical exercise can do for if you have a heart problem or you have any other types of problems. We start knowing and quantifying by, excellent people in epidemiology or urban, the importance of green areas in cities, as compared to cities that do not have green areas. This is something that is starting, or water places, etc. And for different reasons, sometimes we don't exactly know the cause and the mechanism, but we know that it is much better. And in epidemiology, you can quantify to a certain extent these associations. So it is starting, but I tend to agree that maybe it's certainly not enough, and we should focus more on these, on the good environment. Yeah, sure, certainly.
Thanks, Robert, for your talk. That was brilliant, as usual. Just one question, just citing what Carmen said in her speech. How would you associate in this kind of research end users?
Well, I mean, of course, you can associate them in many respects. I think the environmental health field is a place where, first, you can associate citizens in many research areas. And if I take the example of diet and the best diet and, all these internet, surveys that are done and where citizens, have an input, I think that's a way also of involving more people. The same for some of these ecological studies that are done where people can provide an input. So this is a field where it is possible to have citizen science integrated within more academic or science done by professional scientists. Of course, you have to be careful because you have to be able to be critical on the data that you have. When you have a lot of data, it's really an issue. But also when I have, this thing, I speak a lot with people that are in pure science, Academy of Science, for example, people, and they don't like any input from policy on, the way science is done or on the projects that are done in science, because they think that research scientists should be completely autonomous, and they are, they know better what is best. And when I speak to them, I have to disagree to a certain extent, but I agree that there should be a lot of free science, completely free, because this is how we discover PCR, and then we see that PCR is so important, but it's free science. But on the other hand, we owe it to the citizens that some of these major issues, I mean, it's incredible that we do not have some major program on climate crisis when we know that humanity may be at risk in some, in maybe tens of years. It seems incredible that scientists do not put themselves at the service of the citizens for this. So we need both, and we have to make sure that we do both. It's not a perfect answer to your question, but maybe we can discuss it a little bit later. I need a beer for that.
Thank you. I see many more hands, but I was warned that we should proceed with the time, so maybe one short question and a comment.
Thanks a lot. Thanks a lot for this wonderful and very active presentation that, by the way, I would like to receive if possible, if you would like to share with...
This will cost you.
I had two questions, but you can choose one of two to answer. I was very interested, and in continue with the question of Arnaud on the end users, because you mentioned the link to the clinics and to the clinicians, so how could you see that? And the other question would be on the role of the industry. Yeah. On all these endeavors, how do you see that?
Well, for the clinics, I think we have to work on areas of the exposome that are highly relevant to the clinics, and it is starting. I mean, we have now, for example, thinking about a liver, working on liver diseases and the exposome. When we started the exposome, it was very general. Now we can focus. I mean, this disease together with the exposome, what does it do? And they are interested in that. I tried, for example, to collaborate with the maternity at the hospital where I am, because, you know, the fetal exposome, and how can they translate it into something that is useful? Like genetics at one point, you know, now genetics is completely involved, and I am at a hospital where genetics is extremely involved, but now we have to introduce the exposure. And I think it's close collaboration with the clinicians that we should do that on collaborative projects. The industry, of course, can be involved at all kinds of aspects. Let me take an example of cosmetics, for example. I mean, you know how the pressure is on cosmetics, and we have to make sure that whatever we are doing is not harmful one way or another, and eventually useful. So I think it's in the best interest of the industry to have something that is good, but also safe. And I think for European industry in particular, if it can have this label on it, I think it's extremely. It will cost the industry to a certain extent. It's not free. It has a cost, but it can be extremely rewarding in the long run.
With that, I would like to thank Robert once more and invite Martin. And I would invite Martin Reihey, who would guide you through the next section of this symposium. 5-minute break.
strict timing for if you can all take your seats now. So I'm going to get started because we're supposed to be strict on time. And in this next session, we're going to talk about infrastructures in Europe. And I'm very pleased to introduce Jana Klanova, who's probably the person who's done the most in Europe to build infrastructures related to the Exposome. She's been active in the HBM4EU project, in some of the Exposome cluster projects, and of course she's the one who's been pushing forward the IRENE. So thank you, Jana.
Thank you, Martin. Does it work? Can I move it forward? Can I move it forward? It doesn't work. Can I do something with it? Ah, okay, good. Very good. Thank you. So good afternoon again. We've heard a lot from Robert Baruchel, and you already understand how complex the problem of human exposome is. That we want to look at multiple exposures, looking at contaminated water and food and consumer product and indoor environment, atmosphere, throughout the lifetime. But at the same time, we need to look at multiple biomarkers to fulfill the concept of both bottom-up and top-down exposomics. We need to develop the analytical capacities. So talk about what it means to develop the analytical capacities. Talking about bottom-up exposomic, it means we need to measure the chemical compounds in multiple environmental matrices to be able to identify the exposure routes and find out how much those different exposures affect the accumulation of chemicals in human body. The other top-down approach means that we would measure not only chemical exposures, but different types of exposure and effect biomarkers directly in human matrices, and trying to find out what is the link between chemical exposures and the health effects. I will borrow this diagram from the HERA project. I mentioned it already. HERA was Horizon 2020 project that was supposed to map the research needs in Europe in the area of environmental health for the next decade. And the outcome of this project was that we have 6 major research goals in Europe. One of them at the bottom are chemical exposures, but there is also climate change and biodiversity. There is the urban environment, the need to better assess the risk factors, but also the need to develop European infrastructures and also develop the new generation of the scientists and educators who can deal with the new challenges. But we also need to realize that chemistry is part of all of them. The chemical exposures is not just the exposure itself, but as I said, it is behind the climate change in one hand, but also the climate change is one of the factors behind exposome. It is one of the factors driving the biodiversity loss. If we want to assess the risk, we need to know to have information about the chemical exposures. For urban environment, pollution of the outdoor air, indoor air, consumer products, they are all part of our urban environment. So we need to be aware that we need to build the capacities that can handle the multiple exposures, multiple chemicals, multiple human and environmental matrices, and they must be high throughput. Plus, we need to coordinate, because thousands of scientists dealing with thousands of problems wouldn't help. If we really want to help the situation in Europe, we have to work in the coordinated way. This is one of the pictures showing what the functional exposomic is. It means looking at the life cycle of the human, assessing the multiple effects throughout the lifetime. And it means that we not only need to build the analytical capacities, but we need to build the good links to existing research capacities in different fields. And this is something that Robert said already in his presentation. We need to link with the European cohort studies, existing infrastructures like SHARE. We need to link with the biomedical resources, medical and clinical studies, but especially with the IT infrastructures and all the bioinformatics support. So if our first priority was developing the analytical capacities, our second priority will be developing the functional transdisciplinary partnerships and approaches. So these needs were already identified in the previous roadmap of European Strategic Forum for Research Infrastructures. ESFRI is actually one of the most important European tools because the ESFRI bodies are mapping and following the research situation in Europe and looking for the new challenges, mapping existing landscape of research infrastructures, and identifying the future needs. So every few years, the new ESFRI roadmap is being published, mapping the current situation, but also identifying the future challenges. And already in 2018, in the landscape analysis in the area of health and food, they identified that we need to start building the European capacities for human exposome. And this is the reason why between 2018 and 2020, we started building the European consortium of more than 50 research partners from 17 countries, not only from Europe, but also from UK and US, to develop the concept of EIRIN as the research infrastructure for human exposome research. Robert, in his presentation, showed that in the last decade, many things were happening in Europe, but many were also happening in the US. This is why it was so important for us to have also US universities as a part of the consortium. This resulted in the submission of the EIRIN project proposal in the fall of 2020. It was positively evaluated and it became the part of the 2021 ESFRI roadmap that was launched last December. You already seen this picture showing that in the heart of EIRIN are those analytical capacities, but it is not only targeted analysis of multiple chemicals in multiple environmental and human matrices, but also we need to use our mass spec capacities to measure at the same time biomarkers of exposure and biomarkers of effect. And because there is no way in measuring all the chemicals in the targeted way, if we really need to search for emerging problems, emerging chemicals, and if we need to assess the toxic mixtures, we need to use the new methods for non-target screening or for emerging problems. This is another challenge for building harmonized approaches to build European capacities. And it is not only about the willingness of the research partners to work together, but we need to team up with the companies developing new technologies, because it was true for the genomic research. The researchers had to team up with the companies developing the sufficient capacities to support the research before we were able to build sufficient high throughput capacities for the genomic research. We are right now at the same phase. We need better technologies, higher capacities, high throughput facilities to support this kind of research. So why Masaryk University and why RECETOX is the one who was leading this initiative? This is because we were 10 years ahead of other partners in Europe, because RECETOX is the European Center of Excellence for Human Exposome Research that is supported by the Horizon 2020 widening action and 30 million euros investment into the teaming project. The teaming projects of Horizon 2020 is the research scheme or funding scheme that is supposed to connect the partners from new member states and the more developed ERA region. And in our case, we teamed up with University College London and ETH in Zurich, but also some existing ESFRI infrastructures like BBMRI to really build the European Center of Excellence. Of course, it didn't come out of nowhere. It was building on 30 years of history of RECETOX as the Environmental Health Research Institute. And for us, it is very important to link those three tops of the triangle. It means looking at the environment, the human individual living in the environment, and also the society that is really creating the conditions for the individual to live it, but at the same time exploiting or affecting the environment we are living in. And it is very important to connect all those domains in order to solve the problem of human exposome. The advantage of the RECETOX Center is that as a part of the structure, it has RECETOX research infrastructure that has been in the national roadmap of large research infrastructures already since 2010. And we were building this facility gradually. So at the beginning, we built a cluster of the accredited laboratories capable of measuring the targeted chemicals both in environmental and human matrices. Then we developed environmental monitoring networks. In the next stage, we supported the population studies, looking at the different populations and ages. And we also invested a lot into development of the information portals and information systems. So currently, RECETOX is behind several global atmospheric and aquatic monitoring networks covering several regions. We developed the information systems that are supporting both environmental program of United Nations and WHO in collecting the data for a global monitoring plan of Stockholm or Minamata conventions, but we are also running several population cohorts. We do have the grown-up birth cohort that was recruited in 80s. We are currently collecting the new birth cohort, but via our collaboration with University College London, which has the research interest in Central and Eastern Europe, we also are responsible for the happy aging cohort in the Czech Republic, Poland, Lithuania, and Russia. So linking all those resources together gives us a lot of opportunity for a combination of the environmental and population studies. But again, in the heart of it is the laboratory capacity. And in the last years, we invested a lot into building not only the laboratories for targeted analysis, but also for targeted and non-target metabolomics, so combining the targeted and non-target approaches. And I believe this is the right combination, because you can't only do the non-target screening, because for the regulatory purposes, you will always also need to have some targeted data. So it's very important to develop both sides of the capacities. If you look at those pictures that were taken from the current roadmap, ESFRI roadmap 2021, those are the diagrams that are mapping the landscape in the domain of environment and the domain of health and food. You can see how the new capacities built by ARIN infrastructure are important and how they are listed in numerous subdomains, because those analytical capacities are important for mapping the atmosphere, water, food, to be considered when we are looking at different clinical studies and so on. But there is one more aspect that shouldn't be forgotten, and it is the education. I already spoke about it previously, that we need to develop the new educational programs. And this is where EDIC University Network can contribute. It is very important, and we are discussing it a lot in ARIN now, that we need to somehow harmonize our educational capacities. We are working on harmonizing our summer schools, building the joint PhD program, and currently I've heard you are also discussing building the joint master exposome programs. And this is again something where we can contribute a lot. And the last thing I would like to mention is that the research facilities or research infrastructures also have a very important regional aspect, because many governments would ask why we should invest into building capacities that will be serving internationally, not just nationally. But such capacities are always also serving nationally and regionally, because you need to be in touch with the common citizens, with the regional authorities. And in our case, we built a very good relationship with the regional authorities and the South Moravian Innovation Center. We are an important part of the regional innovation strategy. And we, together with the regional and city authorities, are building something we call the Partnership for the Healthy Future. We call it a Brno Living Lab, and this is the place where the science meets the regulation and common citizens. So I hope that next time we will meet in Brno. It's not really the mining region. I think we are closer to Vienna than we are to Ostrava mining region. It's actually the city that was famous to be Moravian Manchester, city of textile industry, machinery, military industry. Nowadays, it is especially the city of science. There is a lot of research hubs, and also IT city. Today, I think something like 60% of all electron microscopy is produced in Brno through the Thermo company, and about 80% of all antiviral software is also produced in Brno. It's also the city where Gregor Mendel worked, and if you come this year, we celebrate 200 years from birth of Gregor Mendel, so you will be able to see a lot of activities. And this is the legacy we are linking to, because he developed or put the background for the genetic research, while we are really contributing to mapping the non-genetic part of our research. And your best opportunity to come to Brno is ICRI, which is the International Conference on Research Infrastructure that will be organized in Brno under the auspices of the Czech presidency in October. And we are planning to organize the side event that will be focused on ARIN and how ARIN should be this missing link between existing infrastructures, contributing the chemical exposomics into existing research infrastructure and building those functional blocks. Sorry to be too long.
It's okay. I think it's best to take the discussion. Well, thank you, Jana. We'll have the discussion at the end of this session. Okay, we'll take it all together, the infrastructure questions. I'm not going to introduce myself. So, merci beaucoup pour l'invitation. I'm very happy to present the European Human Exposome Network, which I think is a very relevant infrastructure here to discuss with you. I work at IS Global in Barcelona, and I am the coordinator of one of the exposome projects that's currently funded by the Commission Athletes. And I coordinate the network currently with one of the other projects. We have a rotating coordination of the EHEN. And I want to give you a brief overview of the network, the type of activities, the type of projects especially that are funded within this network, the things we do together, which is mainly working groups and some other activities, and then some short points on the infrastructure needs related to the exposome in Europe. So who are we? This is EHEN. This is the European Human Exposome Network. These are 9 projects that were funded under Horizon 2020. They all started in January 2020. 9 projects with partners from 24 countries, 126 research groups, and the funding is around 106 million euros for the 9 projects combined. And yeah, the Commission has basically asked us to work together, which is, I think, a really positive development in the Commission at the moment, is to group these type of projects together in clusters. And the Exposome cluster is one cluster. There's other clusters on other important topics or topics that are very much linked to the exposome, like the endocrine disruptors, the microplastics, etc. So we are 9 projects, and 9 projects that are all focusing on different aspects of the exposome with some overlaps as well. So I've got a few slides to try and summarize that. Some projects focus on certain parts of the life course. My own project, Athlete, focuses on the first 2 decades of life as one of the vulnerable life periods. There's one other project that focuses only on the early life period. Then there are other projects that focus on the entire lifespan, but on a more one specific disease outcome. For example, the Remedia project that focuses on respiratory diseases. There's 2 projects that focus on immune-mediated diseases. They cover the entire lifespan, but just one group of diseases. Then there are projects that focus more on certain groups of exposures, such as the urban exposures that are covered by the EXPANSE project, for example, or the occupational exposures covered by the E4 projects. So you see there's quite a range of different focuses in these projects. What they have in common, I guess the projects are all trying to implement the exposome approach in that they all try to focus on multiple exposures from different exposure domains, looking at all the different levels of the exposome. As we've heard in the previous talks, there are many different levels in our exposome, going from the molecular, the individual personal exposures, to the ecosystems. So some projects try to cover many domains, others are very much focused in one domain. This is trying to summarize a little bit where we are with the different projects. I've probably left out a few of the things. The social part is covered in a lot of projects, but there's 2 projects, for example, that have a stronger focus on the social exposome. Several projects include the urban exposome as one of their main focus areas. Lifestyle factors, diet, physical activity, etc. are covered by quite a few projects. Then there's the chemical exposome, which is covered by various projects in various degrees of detail. There's projects that cover more the occupational exposures. There's projects that cover more indoor exposures. And then there's Athlete and EXPANSE, I think, where those are the 2 main projects where we integrate the non-targeted chemical exposome that we saw in the last 2 talks. In Athlete, we have a complementary approach to having both targeted and non-targeted coverage of the chemical exposome. And then there's a large, in quite a lot of the projects, there's quite a large focus on omics methods, because for an exposure to have some type of health effect, it has to have some sort of biological response. Yeah, there's no other way you can get to a disease. There have to be biological responses, and if we can build up better knowledge of these biological responses, we can do a lot more to try and understand the diseases, to develop biomarkers for the diseases. related to the exposome. So again, this is not an exhaustive picture, but various projects focus on different layers of the molecular exposome, or the internal exposome, if you want to call it. The epigenomics, for example, have been mentioned as one really important possible mechanism by which the environmental exposures may impact on our health. So that's one of the layers covered in various projects. So I hope that in a few years' time we will get a much better idea of how far we can go in these population studies, because these are mainly population studies, not so much toxicological studies, in integrating the molecular and high-throughput omics methods to better characterize the exposome, but also the biological responses to the external exposures. So there's a lot of data being built up in these 9 projects. And the activities that we do together are mainly centered around making this research visible to the outside world, through the website, through meetings, through newsletters. We have publications for each of the projects that have been published now in the last few months in environmental epidemiology. But then also through internally working together to exchange methods, to work on the communication and dissemination, so really making the links to the stakeholders as a network, so that this type of research becomes more visible, but also so that the people, the end users, so to call them, understand better what we can do with exposome research. And I think this is an important bridge that we really have to manage. We have to cross that bridge between our exposome research and how we explain that to the end users. There's a group on ethics and law, and then there's a really important group on metadata. I will talk a little bit more about the metadata group, since we are talking about infrastructures here today. So metadata is a working group within the EHEN, and the aim of that group is to work towards metadata verification in EHEN. So I assume that most of you will have heard about FAIR data. FAIR meaning findable, accessible, interoperable, and reusable. So this is key in trying to pull data together and trying to make sure that we can find all the data, even after the projects are finished. This group is led by Morris Swerts, who's a partner in 2 of the exposome projects. And these are data scientists who are setting up platforms that help to make the data findable, first of all. So as part of the EHEN, we're working on collecting metadata from all the projects and to turn that into a findability catalog. Now, findability means, we need to be able to find which projects has which data, where can we find it, how can we access it. To catalog all that is already a big job. Yeah, so this is one of the first steps, is actually making data findable. And this is a platform that we're trying to build on now, because each of the 9 projects when they started, had somewhat different approaches. We're working now with a Molgenis platform, which is one of the platforms that is used, that we've been using. We've been using this for a few years now with the birth cohort projects. And here we're trying to put all the data, the metadata together in a harmonized, interoperable form, so that we also use the same terminology for the same variables, so that we use the same names for the same variables. So this is a long process. It's a lot of harmonization work by the individual studies, but in the end, we should work towards a data platform where people can go, they can find the cohorts, they can find the studies, they can find the exposome data, And hopefully also there will be some level of harmonization of the variables that are there, of the terminology that's used. So there are now EHAN subgroups that are working on these issues related to the clinical data in the projects, related to the chemical exposure data in the projects, and related to the omics data. And they are coming up with proposals for some degree of harmonization, common code books, and metadata ontologies, as these are called. This is a lot of work, because these are 9 projects that were funded to do their own work, but now are also working together to try and put together some level of, as we call it, interoperability. And 5 years is not an awful lot. It looks like a long project, a 5-year project, but to work on this level of metadata it takes time. So, where are we? Well, we're building this strategy. This is the type of work that these groups are doing. First of all, making inventories of all the types of data that are available, the type of vocabularies and code books that are used in the different projects. I won't go into this for too much. Building a catalog. So this is a findability catalog. It's just to be able to find the exposome studies. So what we're doing in Athlete and some of the other projects is actually documenting the cohorts in these type of catalogs. This is the catalog that's built by the people in Groningen. It's a really important aspect of FAIR data is the findability catalog. As I said, we're working on interoperability. This is the type of work we've been doing with the birth cohorts, where we have been harmonizing all the variables. And we are also, especially from the part of ICE Global, adding on all the environmental exposures to these catalogs. This is, at the moment, focused on birth cohorts, but we really hope that others in the EHAN will pick up these type of catalog platforms to do the same, so that we have, in the end, some commonalities throughout the exposome projects. And yeah, this is the type of work we're doing in terms of harmonization, so that if you have, there's other ways of adding omics data to these catalogs that we're also working on at the moment. And then, apart from that, there's people working on the access and reuse issues, setting up systems for federated data analysis. So this is somebody accessing cohort servers from, remotely, but without being able to access individual data, so that we don't run into problems with the GDPR. So these are systems to analyze data in a federated way, where only the summaries of the data and the metadata are exposed, but you can still analyze it using all the standard epidemiological models. So there's a lot of work here on building these type of analysis methods as well. And in the toolbox that we saw earlier, all 9 projects have as one important aim to build an exposome toolbox, that can be used after the project finishes. So one thing that EHAN wants to do is to try and put that toolbox as one virtual searchable toolbox available on the website, so that people don't have to go to 9 different projects to find tools, because that would be a mess. And these tools are in many different areas. There's tools related to the data, as I just showed you, in terms of metadata, but then there's also tools related to exposure assessment protocols, data analysis methods, bioinformatics methods, risk predictions and risk scores, impact assessment. We haven't mentioned impact assessment much today, but I think that's an important tool for translation to policy as well. If we can put the impacts on health into numbers related to many exposures in our environment, that can really help us to translate some of the exposome impacts. Intervention, several of the projects are setting up intervention studies. So the idea is that we will have, by the end of the 5 years, a common toolbox between the 9 projects. So, as a last slide, some thoughts on infrastructure. I think obviously it's going to be really important that we talk and that we coordinate closely with the other infrastructures in Europe, especially IRENE and PARC. I think that's it's going to be really important to build the links between these 9 projects and these other big initiatives that are starting now. In terms of data, I think we need a good infrastructure that can last beyond the 5 years, because this is a lot of work to build up. By 2024, the projects will run out of money, and we might just be at a point where we have a metadata infrastructure where the data is findable, but that needs to be maintained. Yeah, we will have, what I hope very much, we will have a databases with targeted and non-targeted chemical data in longitudinal cohorts, with the external exposome, the urban exposome, the lifestyle, the social factors, with the clinical data, and with all these omics layers. And it's really important that these type of infrastructures are maintained. I see the hand. So I think this is where our real need is to maintain the infrastructures, and that these things don't collapse after a 5-year time span. And then in terms of infrastructure, the toolboxes are important, I think, as well, so that we don't lose the common protocols and statistical analysis and bioinformatics methods, etc. I'm going to stop there. I think we will take the questions after.
Pascal Sanders, yes. So, oops. Thank you for the invitation to be here. I am with friends from Brittany, from France, new friends from Masaryk University, so it's a nice day for me to be here and to present PARC. So PARC is a partnership for the assessment of risk from chemicals. I am the scientific coordinator, and it will be coordinated by a team at ANSES with the participation of all our partners. I am happy also to have chosen the same iceberg than Robert. So he explained everything, so I have nothing to say, only that at the European level we have the most developed system to the assessment of chemicals with different national agencies and also the European agencies working together to develop the assessment in the area of food safety, but with ECHA we have a measurement of many properties of the chemicals. I will say also that I am a baby boomer, as explained by Robert, and if you know the risk assessment of chemicals start a new era after the publication of the Silent Spring by Mrs. Carson, and the Environmental Agency in US have changed the process to perform this risk assessment with the concept of hazard assessment, of risk assessment, and exposure assessment. It was a very big step in the way that we have developed the system to assess single chemicals, and we develop at the global level a new system for the assessment of the risk. But in the meantime, the industry invents a lot of chemicals, and we have during the last century created a lot of new entities and chemical entities. And if you have a look on the global market for the chemicals at the global level will double between 2015 and 2030. So the pressure on the environment and the pressure on the health continue to grow due to the development of the humanity, and it's something very, very important to have in our mind. And you know also that 2 weeks ago we have a publication that we have reached, and we are above the limits about the new chemical entities as boundaries for the global development. So this problem is well known at the global level and at the European level. The second problem that we have is the fact that during many years we have an industry based on the mining, use, and waste. And unfortunately, this principle of economic development was also the source of a large pollution in the different areas, and our water system, our soils, our air are polluted, as explained by Robert. And we have to now change our ways to manage the waste and to develop new systems and to change also our usage of the chemicals in our different activities from our daily life and for the industry, and to reach one target at the global level, to have a better life and to have to live in the park as explained by Robert, with a low exposure to chemicals. So in the Green Deal, the Commission, the new Commission with the member states has explained that we have a new chemical strategy for sustainability towards a free and a toxic-free environment. And the idea is to protect human health and the environment together to contribute to a non-toxic environment, which means that we have to reduce our exposure to chemicals of concern for health and for the environment, to minimize our exposure, and to develop a way to have safe chemicals for any use, non-necessary use that we have in our life. It's a big challenge for the people in charge of the risk assessment for chemicals, but because they have to define what is safe and how to assess the safety of these compounds. Then we need innovative tools for testing and risk assessment, and one of the reasons of the establishment of PARC is to develop these new innovative tools and testing for risk assessment. Another way to present what we have seen with the exposome concept is the fact that for the risk assessment of chemicals, we will take together many information about the chemicals, about the hazard assessment of these chemicals through different kind of studies, from in vivo studies, in vitro testing, and from epidemiological studies or environmental studies. We will apply a way to understand what is the hazard, and the adverse outcome pathways is in our new tools that we can use for the hazard assessment. We will take also into account what is the exposure, and the exposure assessment, as explained by Robert, is a combination of different information: how we use the product, what kind of consumption we have, what kind of usage we have in our daily life, what kind of usage we have by industry. We have to monitor this exposure also, and we have to know also the toxicokinetics of the drugs, and we have also a lot of studies to perform to have this information, or to use any system in silico system which can be transform information about the chemicals as information about the toxicokinetics, for example, or the hazard assessment, which is named the quantitative structure-activity relationship to model the hazard or to model the risk. So it's important to have these tools for the new generation of risk assessment, and it was one of the reasons of the organization of PARC. What is PARC? It's a public-public partnership under Horizon Europe. We work with a lot of people coming from HBM4EU, but also from colleagues working in different research projects to develop this public-public partnership. It is planned to work during 7 years with a budget of 400 million euros, which will be confirmed 50-50 by the European Commission and by the member states. We have now 28 countries participating. It is a network of 200 partners, so it's a big deal to organize the things. But we are very proud to have present in this network of partners 3 European Union agencies: ECHA for the chemicals, the European Environment Agency, and the European Food Safety Authority, which is very new to have this for ECHA and EFSA, the participation of these 2 important European agencies, but we have also the national agencies working on the risk assessment present in PARC. So we have Institute of Research, universities, and these agencies. How we organize PARC? It is organized according to very specific objectives. The first one is a high-level network for the regulatory science interface. We have in the governing board of PARC members from the 5 DGs, European DGs, RO, Santé, Agri, and Consumer. I don't remember all the names. We have also established a strategic research and innovation agenda to move towards the next generation of risk assessment, and it is organized with 4 work packages, one about the monitoring on exposure, and I will develop this work package activities. The second is about the hazard assessment. The work package 6 is about the innovation in regulatory risk assessment, and we have to develop the concept and the toolboxes necessary for the application of the transition to the new strategy with the chemicals, which will be organized by work package 8. And you can see that we have also 2 work packages in charge of capacities and platforms. As explained by our colleagues about EN, it is extremely important to develop the concept of fair data, because all the things that we have discovered as data and create as data needs to be findable, accessible, interoperable, reusable for the risk assessment, because it's our source of information to perform any kind of risk assessment. The second, as explained for Irene, is the fact that we need to also develop and to train the people, and this is the development of capacities, but we have also to strengthen the capacities of the laboratory networks, and one of these important things is to develop the quality assurance system which is needed for any kind of measurement. So you have here an idea of the origin of the different work packages there's in PARC. You can see that we have they come from agencies in charge of public health, agencies in charge of risk assessment, agencies in charge of the environment, and we have also people coming from university or for research institutions. And you can see that Masaryk University is present in work package 9 in charge of the infrastructure, so we have a good connection at this level. So I will continue with more precisely. Well, in 1 or 2 minutes, so it will be very short. I can continue. So we have, in this work package, our colleagues from UBA from Germany and for France, Santé Publique France. I speak on behalf of Sébastien Denis, because Sébastien is the leader of this task, but unfortunately cannot be with us today. In this work package, you have 3 tasks. One about the human biomonitoring. Then they will continue the work done with HBM4EU, and the managers of this task come from from HBM4EU. The second level will be in charge of the environmental monitoring, and we consider that it is important to understand the presence of chemicals in the environment and to use this data for the measurement of exposure. And the third task, which is managed by our colleagues from INRAE and from the French University in the Netherlands, will be about the innovative tools and methods. So the work package for the monitoring and exposure is very important because it's the biggest budget with 120 million euros. So I will say they organize the things that they will cooperate and collaborate between the 3 tasks to develop the system of monitoring and measurement of exposure based on the development of the innovative methods, and they would like to foster the collaboration between the different networks existing at this moment in Europe in the different domains. So for the... I have a little more time. The reason to have friends in the organizing committee. Okay? It's not ethical, but it's practical. So we have a plan for the human behavior monitoring to continue to work as it was done in the HBM4EU. So we will organize a general survey, and they have planned to organize some few targeted surveys about, for example, the dietary exposure of children, or about the newborns with a breast milk survey. They will also work on hotspot residents, for example. They will also combine this with some case studies, some add-on studies where they will use new methodologies for certain type of measurements, like the use of the non-targeting screening or the suspect screening in some part, in the investigation in some part of the population, and they will also perform some targeted analysis for a list of chemicals that was decided in HBM4EU. So they will combine these studies with occupational survey, and they plan from the beginning of PARC to work with the waste management workers and also with the healthcare workers, because they are exposed to different type of chemicals. And then they will continue to work on the sustainability of the surveillance system for Europe, and in close collaboration with Irene, I will say. So here you have more details about what is planned for the beginning of the task 4.1, and I will continue with the task 4.2. At this moment in Europe, we have different systems of monitoring of chemicals in the environment, because we have the Water Framework Directive, so we have a lot of measurement. We have also measurement in air, as you have explained. We have to develop something for the soil, because we have a lack of data about the pollution in soils. And they will start by the setting up of an overall process of environmental and multi-source monitoring. And they will start with two case studies, one about PFAS, because it's a hot topic at this moment for the European Commission and the member states, and they will measure the background levels after the use of decades of use of these chemicals, and they will also work on the endocrine disruption compounds to establish by these case studies this system of environmental and multi-source monitoring. And based on what they have, they will try to expand the system to new media and to other matrices and compartments for both class of chemicals, and then they will develop the system for other class of chemicals during the implementation of PARC. And for the task force 3, which is under the responsibility of the analyst, they will continue to develop the new methodological approach to the available with innovative personal to community sampling methods, to have information, to have new sampling methods, to develop new methods with improved sensitivity, because in the field of chemical exposure, one of the problems is the very low concentration that you have to measure in different media. They will work on the effect-based monitoring approaches with the combination of bioassays and effect direct analysis to link the exposure with the effect, and continue to develop the suspect and non-targeting screening through the application of this method in different case studies. And it will also important to continue the development and to continue to work on the different challenges in the chemical risk assessment. But I will say it's the same for the people working in the exposure domain, and we will share the expertise and discuss how to go to a system more standardized and under quality assurance system for the future. Thanks for your attention.
Thank you very much. Thank you very much. We're going to ask for questions for all three of the talks, especially questions that link the three of the talks. So there's a question over there.
Yeah, thank you very much. I'm working for the research infrastructure program, and I've also been working for health research before. And I mean, the point I wanted to make is it has to be very nice to see these different initiatives together.
Is there another microphone? Because we can't hear very well. I think there's one here.
Thank you very much. I think it's important that the audience could hear you too.
Thank you. Thanks. So I've been working between the research infrastructures and the health research program, and from that angle, I'm very grateful for this sort of nice presentation to have these initiatives coming together. You have this EHAN network there that very clearly sort of spells out the needs, and we nicely heard this sort of data needs, yeah, this sort of issue as a priority also in your partnership there. I see relatively little of this in the infrastructures effort now, because I think it's really focused on the analytical facilities and you know, high throughput and lots of things, but there's a reference to the share cohort or something, but I would see a major activity needed to assemble in a structured way the cohorts to have the data resource that you want to feed in chemical stressors into them. So I think there is something that I would feel would deserve to be taken up by the infrastructures effort there. That would be one point. And the other thing also, I mean, you are now working across the EHAN projects on the metadata that is a low-hanging fruit. So I mean, to get the harmonization of the data variables there sorted out, it's another big effort, and there's a lot of parallel activities. And we had this workshop, or you organized that workshop in 2020 on the data, or it was a different environmental health project, but EHAN also was present there. And one outcome was that I think there is a lot of activities ongoing already for the European health data space. There's BBMRI who takes a role for many of the sample-linked data there, and I see little from that in the infrastructure. So I think it could be worthwhile to consider to take that up into the remit there.
I think Jana can take that one. Yeah.
Yeah. Sure, I absolutely agree. You have to. Sure. Sorry. I definitely agree, but you have to consider that EIRIN is something that just happened to be in the roadmap. So pretty much it has about 10 years to be fully developed. We went through the design phase. We had to prepare the project. Now we will have 5 more years to go through the preparatory phase, which means we really need to work hand in hand between the European level and the national level. And this is one of the very important messages from today, that nothing can be really built from the European level. European level is a coordination. We convince them together that this is important European priority. We can create consortia, write some European projects, but the capacities must be built at the national level. It is very important. And here we have to use everything that is already there. We are talking to our partners, mapping existing capacities, but also working very closely with existing projects. And I think that this is what Martin said in her presentation, that they will be developing some new tools in the EHAN project, but hoping that at the same time we will work with them to put all those results into this new European infrastructure to provide the future and sustainability to all those tools. And it is also why in the PARC project we have 2 work packages that are infrastructural. This is exactly the reason, because PARC is one of the financial tools that will support building the European capacities. So one of them is work package 7 on data. So it's linked to EHAN and other existing projects. We will be working together to develop the common ontologies, but also working with existing infrastructures. And I mentioned that we want to organize the side event next to the ICRIC conference later this year, and this will be discussion among EIRIN and all the existing infrastructure. We already have an agreement from BBMRI, Elixir, SHARE, Actris, ICOS, environmental health and IT infrastructures, to make a future plan, how we will proceed with linking all the data, because we can't assess Exposome without bringing together social, health, environmental, and all kinds of other data.
Let me just add that the cohorts are absolutely fundamental to this. And these are long-term studies that have been going for decades, and they've been collecting exactly that type of data, the social factors, the lifestyle, the environmental exposures, whether it's green spaces or air pollution, or now we're building in the chemical exposures. This is what we need to do life course exposome research. And we need support and maintenance of these type of infrastructures. So.
Maybe one more note. When we were explaining to ESFRI why we need a urine infrastructure, we pointed out that none of existing infrastructure, not a single one, is assessing chemical exposures. We have all kinds of environmental and health, none of them looking into the chemicals. So that was the unique part. And this is why I said that our first priority is really building this chemical capacity, because this is the missing piece. But then I said the second priority is really to bring this element and introduce it to all the other infrastructures that can potentially use it. We don't want to build the new cohorts. We want to link with BBMRI and other infrastructures and projects that are already having it, but then adopt their tools, bring them to the common ground and work with them in the future and invest them into the future projects.
Yeah, I totally agree with the sustainability of the cohorts, the sustainability of the data. I mean, we cannot start these things if we don't have something that, we should really make them sustainable in the future. It's really critical, I mean, for everybody. And it's a toxicologist that says that, so
it's really good to hear.
No, I mean, there's no question about that. I think we should do something about that. I have a question for you on the EHEN cluster, because there are a lot of activities that are cross-cutting between the different projects. Do you have, it's a really maybe a specific, do you have a specific budget in EHEN for these cross-cutting activity, or how do you do that? Because it has been a problem in other clusters.
We don't have we didn't budget this in. Well, the projects have set aside a certain amount of money within their own budgets to work on the cluster activities.
From the start, okay.
Well, yeah, but it's actually a chunk of money that was in the original proposals assigned to the research work that has been converted to network activities. It's not much, I must say. And that's the one thing we're struggling with is the network activities come on top of the research. There's a lot of hardcore research in these 9 projects that I haven't really mentioned, but that's the largest part of the projects.
I think.
And the other point for me is that the metadata work and everything is really important, but it takes a long time to get these type of inventories, harmonization, etc. And it's only worth for people to work on that if they see that there is a future. So in the EHEN, we really have to give people some perspective of what the possibilities are for the future, because to say, okay, we're going to do all this work and after 5 years it finishes because we run out of money, that's just not good enough. And I think that's where we can all think together what the steps forward are. But I think that's to keep people working on these type of issues and the metadata and all the data science and the platforms, which are, we all agree that it's super important. We need to find a way forward.
I think there is one more important resource that hasn't been mentioned here today, which is EOSC, European Open Science Initiative, because this must be very closely linked to it, and it is another resource that we should bring to support our activities, because this is exactly it. We are today most of the time talking about how we will bring together data from cohorts, infrastructures, joint programs and such. So this will be very important. And this is again homework at the national level, because we can work at the European level, but at the same time you need to be active at the national level, because right now the national EOS consortia are being developed, they are discussing the structure of the data, and we need to be in the middle of it.
I'm getting signs that we need to stop here.
Oh.
Sorry? We go to the next session, or we? It looks like one last comment, please.
Just one comment. One point has been mentioned by I think all 3 of yours. It's toolboxes. We have spoken about data, we have spoken about infrastructures, but maybe it's my background as a physicist that the validation of toolboxes, because what's after is assessing data, assessing risk, assessing exposure that are of significant for public health outcome. And then is the question of, if you want to share the toolboxes and pass it around between the structures from EHEN to PARC, for instance, the question of how these toolboxes are validated, for example, for toxicokinetics models and so on, is maybe a question I would be interested to go on discussion, but maybe later, because the time is probably short now.
For later.
I might just say one very quick comment. Toxicology is the new field, and one thing we need to consider is whether there isn't a place for another research infrastructure on toxicology, or whether we are capable of linking it to IRENE or embracing the field of toxicology, because it's such a huge infrastructure that is needed, that this is something we will be discussing a lot in the next few years.
Okay, thank you to all the contributors. So our next session is more of a roundtable session. We have 4 speakers with somewhat shorter talks, followed by a discussion again. So I would like to ask the speakers to keep it short, so that we do have time for this roundtable discussion. And this is about investing in the infrastructure to respond to the need for innovation in characterizing the chemical exposome. So I would like to welcome the first speaker, Agnès Robin. Good afternoon. Yes, please.
Good afternoon. So I'm Agnès Robin. I have to apologize because Lia Karamali, my head of unit, should have been here today, but she had a last-minute commitment that she could not change. So I'm Agnès Robin, I'm head of sector for research and technology infrastructure policy in the Commission in the DG Research Innovation. And Andreas, who spoke a few minutes ago, it was my former colleague. So sorry, I should have moved to this one first. I just want to briefly explain the context in which the framework that in which we are working now for research infrastructure. So you might have heard about European research area. So it's not a new concept. It was the idea of having a free market for researchers, for knowledge, for infrastructure, access to infrastructure, and so on. So it's still in the air. It's even more. There is a renewed approach for the European research area, with in the last council in November, adoption of a pact, a recommendation for a pact for research and innovation, where we are countries are sharing values and principles, agreeing on some priority areas that they will work together. There is also a discussion for a new governance for the European research area, and I don't want to enter into detail, but from the highest level, which is the Council, to ERAC, which is a high level steering body, and more concrete, coming to more concrete ERA forum, which I would like to mention because there is a strong willingness to engage all possible stakeholders in this ERA forum. And just a few words about ESFI, European Strategy Forum on Research Infrastructure. So this group, this expert group with representative of research ministers is still there. It's not per se a group of ERA forum, but it's working like a group of this ERA forum. So it's just to explain the level of the different governance for the European research area. And the third block is the ERA policy agenda, and here we are coming a bit more in concrete with a list of actions that have been proposed and are currently discussed by member states and by stakeholders. So that's the framework, the policy framework. There are a number of policy actions, and I will just mention the research infrastructure 1, action 8, there are 20 in total, which is about strengthening sustainability, accessibility, and resilience of research infrastructure in the European research area. So Jana has very well described the process with ESFRI, the landscape analysis, and that had identified some gaps. So this we are entering a new cycle with ESFRI, and we will try to have a more strategic analysis of the European research infrastructure landscape, going deeper, because until now there was a general analysis of the ESFRI landscape, of the big scientific challenges, but there is the idea to be more strategic, to have different analysis of the different levels, including not only the infrastructure, not only the scientific needs, but also going up to the level of maybe scientific services offered by infrastructure, both at European level and as much as possible at national level with the involvement of the countries. You see a number of bullet points, so we are working on trying to promote access, in particular transnational access, making all infrastructure available to all possible researchers based on excellence and based on the needs. We are also in this policy agenda, of course, we have the ESFRI roadmap. So ESFRI roadmap is not only about listing the priority infrastructures like ERN, it's also about monitoring their performance. So they have worked on the key performance indicators, and they will follow up these infrastructures, their implementation, their operation, and guiding, working with this infrastructure to ensure that they can deliver what they are expected to deliver as service to the scientific communities. We are referring also to the ERIC framework. So what is ERIC framework? It's a legal framework that has been created by its regulation of the Council. So it provides a legal framework for pan-European research infrastructure. So you can have different legal forms if you want to create a legal entity for a research infrastructure. The ERIC one was one which was created specially for that purpose. And now currently on the ESFRI roadmap, so maybe there are 40 projects that are 40 research infrastructure on the ESFRI roadmap that are either fully operational or under implementation. And among them, half of them have adopted these legal frameworks. There are 23 ERICs. The last one was ANAE, analysis of and experimentation on ecosystem research infrastructures. The last point is we have very much discussed, I've heard about that very much today. It's about increased cooperation between research infrastructures, between research infrastructure and infrastructures, including EOSC, and with stakeholders. So that's really something which is more and more important. Now we have an enriched landscape of infrastructure at European level. There are, I think now on the ESFRI roadmap, there are 63, maybe 41 under implementation, and 22 projects, including the very last one like ERN, that are in this roadmap. So the landscape now is a bit many infrastructures. So the idea is to make this infrastructure, to consolidate this landscape, make sure that there is no overlaps. It's good to have some overlap, but not too much. Making sure that all possible synergies, interoperability, connection and coordination between this infrastructure will be developed. I just want to mention that there are other type of action in this ERA policy agenda, and here is just the example of one targeting universities, higher education institution. We don't see everything. Okay, I will go. I want to see all of them. So just very briefly, the main actors at European level for the policy on research infrastructure. You have ESFRI, which is developing the policy, kind of incubator of strategic research infrastructure, and also working on the coordination of national priorities, and well known for its roadmap. The Commission is also, it's actually a member of ESFRI as well, even if its role is different, but it's also providing some support through the framework program, so Horizon 2020, now Horizon Europe, in particular supporting different phases of the development of the research infrastructure, but also technological developments or transnational access, support to EOS and connection of the infrastructure to EOS in particular. I would like to mention as an example per large domain of S3 infrastructure, for example, environment or physical science and engineering or life science. There are large project clusters where you have all this S3 infrastructure in the same project working together for making their data fair and working on the common solution to connect to the EOSC, to avoid that each infrastructure alone do the same thing as the very similar infrastructure. So that's the idea. This is the current roadmap, and I just don't want to enter too much here. A few words on Horizon Europe. So you see this large line of actions, so what we call destinations. And so InfraDev, InfraServe, InfraTech, InfraEOSC, InfraNet. So InfraDev is really to follow, to support the infrastructure in their development from the very preliminary phase, the concept, design studies, to the preparatory phase for those who, like Erin, has just entered the roadmap to build the case. So they enter the roadmap, they are flagged as a priority for European. This is needed for Europe. Now you have to build the case, see how you will be funded, how you will be sustainable, which communities you target, which type of service, what is your data policy, access policy. So bringing all information available to the funders, which are the member states, associated countries. You present your case, and if all go well, they said, yes, we vote for it, we will fund you, and you have to further develop with a legal entity, and we will support you. So that's InfraDev. InfraServe, it's a bit different. It's mainly focusing on funding transnational access or virtual access to the different infrastructure, either the S3 one or national one, networks of national one. InfraTech is more about developing scientific instruments, new tools, new methods, so advancing the services, improving the services. InfraEOSC, it's 2 minutes, please. It's my last slide. InfraEOSC is really in the support to the development of EOSC, and I've mentioned this cluster to make sure that the whole S3 infrastructure will connect to EOSC, and InfraNet is more about network connectivities, and that's really, it's managed by our colleagues from DG Connect, and I think that was all. Thank you.
Thank you very much, Agnès. If there's any quick questions on clarification, we can take one now. Otherwise, we keep the discussion for afterwards. Okay, if not, Vincent, I give the word to you. Close by.
So, good afternoon. So, today my talk is about trying to compare the funding in the US versus the EU in terms of support for research on the exposome. And we'll try to... Okay. So since the introduction of the exposome concept in 2005, as it was mentioned previously, a lot of research started to emerge around the 2010s with different funding from the US and then the EU. And as you can see, both EU and US follow a different path toward the same goal to characterize the exposome, and including the chemical exposome. But the US choose a path towards building capacity and infrastructure and generate a lot of data on existing cohorts. While in the EU, as you will see, it was more focused on funding project to develop new tools and methods to analyze this complex data and ultimately to link exposures to health. I need to go back. So the first project that was built in the US was Hercules Exposome Research Center, built at Emory University and funded by the National Institute of Environmental Health Sciences. And it was supposed to be, and it was to play a leading role in the discovery, evaluation, and application of the exposome concept. So it was the first big infrastructure in the US and also in the world at that time. The goal was also to add investigators to carry out cutting-edge scientific research about the environment. And this platform provided key infrastructure in terms of expertise to develop and refine the opportunity to map the chemical exposome. So as you can see on the slides, at that time, the investigator at Emory was mostly interested in air pollution and chemicals, and mostly used metabolomics, which was then refined as non-targeted exposomics. But it was a similar technology that was able to both capture small molecules, including exogenous molecules and endogenous molecules, and also a significant focus on birth outcomes and reproductive health. And then in 2015, the NIHS also built a large program called CHEER for Children Health Exposure Analysis Resources. It was designed to be a centralized network of exposure analysis services and expertise. And it was open to researchers who want to add exposure in their study, and it was free for them. It was no cost, no additional cost. The CHEER program was covering all the costs related to the extra analysis. So it also supported some pilot study, but most importantly, it offered a very important services, including expert consultation on exposure analysis, study design, and data analysis, and laboratory analysis, including targeted, like we quantify specific exposure, and also the non-targeted analysis, where we want to be more generating hypotheses about some connection between the exposome and the health outcomes. So it was open and targeted for children health studies at the beginning. They also offer some statistical analysis, data integration, and interpretation, and also a repository of de-identified epidemiologic and biomarker data, offering the opportunity to worldwide scientists to access the data and reuse the data. And we can see that it's coming in Europe as well, but it's a lot of work to build that resource. And this program also supported the resources at different laboratories across the US. And in 2019, the same program as CHEER emerged as the next step, and it was focused on not only children health studies, but also adulthood studies. And so it's the same philosophy. It's a centralized network of exposure services and also open to researchers, but at this time, including human study that they include different life stage in terms of exposures. Again, no cost for investigator, and this time they also included some lab capacity for environmental samples. So it's a good combination between the external environment and the internal environment. And again, different labs have been built to provide the capacity for all of these big cohorts. Now in the EU, the first project and the first big program to emerge was under the FP7. So it was 3 projects: the Human Early Life Exposome, so HELIX, which included 6 prospective mother and child cohorts, and did 1,200 measures of exposures of different biomarkers, including PCB, PFASs, phthalates, pesticides, metals, but also the physical environment like noise, built environment. And most importantly, included both external and internal exposome. As it was presented earlier, they also included some molecular profiling and biological pathway to evaluate the link between the exposure and the biological pathway impacted by these exposures. And I think they also did a great job to try to think about new statistical approaches to handle this big data. The other project was the exposomics, focused on different life stages, early life, childhood, and adulthood. And it was also to build new approach to assess environmental exposure, including air and water pollution, and different health outcomes. They also used some omics techniques to evaluate biological changes associated with these exposures. And the third project under this program was the HEALS project, the Health and Environment Wide Association based on large population surveys that try to build child cohorts, including singletons and twins, across 10 EU countries. And they also add some very important information about external and internal exposure, socioeconomic biomarkers, and health effect data. And also build these new tools needed to perform what we call the EWAS, the exposome-wide association studies. And also, you can see that the omics play a significant role to link exposure to mechanistic and adverse outcome pathways. And ultimately, all of these projects provide a new insight on the link between the exposome and several health outcomes. Also, another project mentioned earlier today was the European Biomonitoring Program, HBM4EU, funded under the Horizon 2020. So the goal was to really bridge the science and the policy, explore current question in chemical risk assessment, and we see it's not easy, to take policy measures to reduce human chemical exposure, and most importantly, to try to harmonize the biomonitoring level data at the EU level, which involves 28 countries, and sometimes countries don't use a similar analytical methods. And it was also one of the first projects to integrate this non-targeted high-resolution mass spectrometry to try to map the chemical exposome. And also one of the goals was to understand exposure-response relationship and to assess potential health impact in different age groups and gender, and also investigate biological effect of chemical mixture. Also presented earlier, so it's easier for me to go into more details. But again, we can see over the last slide that the EU invested a lot of resources to build some cluster or network of different projects focused either on different life stage or focusing on different health outcomes. And to summarize all of this information, what we can, like the take-home message would be, all of them, the two different models have their own strengths and weaknesses, and are kind of complementary to each other in terms of how they approach the research on the exposome. So the US built a network of laboratory to build capacity to analyze thousands of samples because one lab cannot handle that many samples. They provided some targeted and non-targeted chemical analysis and provided services to help investigators to analyze their data. They built a data repository to reuse the data and to be open to the scientific community to generate new hypotheses about some link between the environment and health. They supported pilot studies. But in the other end, dealing with these thousands of samples, they did not have time to do more research on developing novel methods to really dive into the chemical exposome. At that time, there were also some limitation on the library used for identifying chemicals, and we can see it's not easy. And also, most of the cohorts, because they were designed before these programs, most of them were not designed for exposomic research. So in EU, all the different projects have included cohorts across different countries. So it's very important strength to develop new tools for measuring the exposome and analyze complex data, use multiple omics, and finally provide new insights into the links between the environment and health outcomes. The weaknesses, it's mostly targeted chemical analysis, but we can see the non-targeted is coming. And also there is limited analytical capabilities in the EU to handle all of these samples. And I will have two more slides. It's going to be quick. So if we can picture, we still have challenges even with that many projects on the exposome, and most of the challenges are how we can capture the chemical exposome. So for example, if we imagine a network of chemicals that interacting with each other, what we can see, the blue bubble currently, what we can see today is only a small portion. It's because there is some hidden chemicals, that chemical we cannot detect because they are present at so many, at a very low concentration, but also because some of them have very different physical and chemical properties. And there is the unknown chemicals, that the chemicals we can detect, but we cannot match or identify based on the current databases. So we need harmonization, and we need to have lab capable of analyzing thousands of samples. And to do that, we need high throughput analysis, and we need high level of networking between countries and between big facilities. And we also need to be more comprehensive in the characterization of the chemical exposome. We need to use complementary analytical platform, develop new technologies and new bioinformatic tools to be able to identify these unknown chemicals. Thank you for your attention.
Thank you, Vincent. We will go straight to the next speaker, Bruno Levizet.
So good afternoon, all. So I will start my talk with an artistic vision of the scope of intervention of my laboratory, so Laberca, National Veterinary College in Nantes, France. This laboratory is strongly involved in the characterization of the chronic exposure of consumers to chemical hazards. So we worked on food and consumer exposure since 30 years, but we moved very early to the environment valency to look for the origin of the chemicals, to understand their transfer to the primary productions, meat-producing animals in particular, and their transfer to a consumer. And then we moved afterwards to the movement of these chemicals in the body of the consumers to try to understand how they were stored, how they are eliminated. And we started at this time, I would say, to try to associate the exposure with some human health outcome. So this, I would say, artistic representations, the interactive triptych of the artist Arman, one of the last sculpture of this artist, is probably a bad representation of the continuum between environment, food, and human health, because till now, I would say it is almost a discontinuum. Historically, the different disciplines worked separately, and it is only a new trend now that all these disciplines are coming together, and I think the partnership park is a very good example of this new movement. So to link the exposure to health outcomes, there is a certain number of data sets which are missing at the beginning. The first ingredient on this slide is probably to provide external characterization of the exposure for us, I would say for the consumers. We did it alone, we did it with the Food and Agriculture Organization, we did it with ANSES in several total diet studies. We rapidly involve as well the characterization of the internal exposure by providing biomarkers of exposure, and probably in France at least among the very first association between external exposure and internal exposure was done in 2008 and 2012 on two total diet studies, the first one involving high consumer of seafood products, the second one high consumer of fish from the rivers. And we really gain in refinement of the risk characterizations by combining the two sources of information. And after we introduce, I would say, observations on the descriptions of biological profiles. And in this example here, I mainly speak about biomarkers of effect, and particularly about metabolomics. And when you merge all these three data sets of information, and of course involving some toxicological aspect like AOPs, for instance, you are in positions to link better the exposure on some health outcomes. So coming back to my laboratory, because it is a testimony for this talk, Laberca is based on three pillars. The first one on historical is a national reference lab involved in the food control, covering a large set of chemical hazards from environmental contaminants, persistent organic pollutants, some food contact materials, as well as a certain number of drugs from phytosanitary products to veterinary products. And we are, for this aspect of the activity, strongly linked to the competent authority, the General Directorate for Food of the French Ministry of Agriculture. The second pillar of our organization is a research lab, what we call in France a research mixed unit. Mixed meaning associations of establishments, the National Veterinary College, ONERIS, and one big research institute, the National Institute for Agronomic and Environmental Research in Haye. We have two axes of basic research, one being food on expology, characterization of consumer exposure, and another axis of research, which is right in here, health, meaning the study of association between exposure and health outcome on better causality if applicable. And of course, I'm going now to the platforms. All the data we produce are coming from two platforms inside. One being on the right side of the slide, metabolomics, so mainly markers of effect. It is strongly connected to an already existing national infrastructure, MetaboHub. On the other one, on the left, HBM for human biomonitoring, more focused on markers of exposure. On this, I would say, platform is part of the France Exposome national infrastructure, which will be developed later. So we are, for the HBM platform, really in connection with the searcher, with the epidemiologist, holders of medium or large cohort, as well with biobanks managed by the health sanitary agencies. So, of course, platform means a certain number of instruments. Concerning our facilities, it's something like 25 mass spectrometers, representing 7 to 8 million euros investment over the last 15 years. The most contributing entities are, for us, the region Pays de la Loire, the Ministry of Agriculture, and, of course, the European Union with the European Regional Development Fund, FEDER in French. So we have three main approaches, and I think the terminology and the semantic has been used already. The first one on historical is probably what we call the targeted approach. I mean, we are focusing on non-compounds, more or less in most of the laboratories, 1,000 parameters. Non-compounds, sometimes regulated. Probably the best approach for quantification, for robustness of the data, but limited in terms of the wideness of the human chemical exposome. So it means that most of the approach are relying on mass spectrometry, either multidimensional or high resolution. The idea behind is strong selectivity, meaning high sensitivity, so low limit of detection and quantification, and at the end compatible with the extremely low levels of concentrations for most of the chemical hazards in human fluids or tissues. The second emerging approach, suspect screening, has been used as wording, at least for the moment. And you can see on the silhouette on the left that the scope of the exposome is enlarged by this kind of approach. And the main idea behind is to generate in silico by the computer an almost unlimited number of possibilities regarding chemicals. I can take the example of pesticide. You may imagine with some software the metabolites, the degradation products of pesticide, the metabolites of the degradation products, etc., etc., so that we can build up some databases up to 100,000 possibilities and more, of course. On the right side of this slide, you can see that with the introduction of ultra-high resolution mass spectrometry nowadays, we're almost in positions to take pictures of the global sample, biological samples, we can say fingerprinting. So we can generate thousands of descriptors, and we have to match the in silico data with what we have really produced on the samples, and according to some dedicated algorithm, some criteria for annotations, chemical structural elucidation, we can match and we can find emerging compounds. So it contributes to the enlargement of the definition of the human chemical exposome on its reality indeed. And the last approach is what we call non-targeted screening. It's something broader again. There is no a priori, there is no hypothesis. The only driver is the toxicity of what you're looking for or the biology of these compounds, either by an interaction with receptors or it can be as well chemistry-driven. On the next slide is an example of a chemistry-driven approach. On the left side, you can see or imagine a kind of profile with thousands of descriptors obtained by ultra-high resolution mass spectrometry. According to a recently developed algorithms were able to simplify these very complex informations, leading to perhaps dozens of informations only. And by this way, focusing here on persisting organic pollutants, we were able to point out new compounds like chloroparaffin some years ago in food and others. And what you can apply here to human samples, we do as well for environmental sentinels, like on seabird eggs or on the adipose tissues of marine mammals. So very interesting to go upstream. Of course, afterwards, we have to objectivize the real exposure of human, but very important information to take upstream. On my, yes, on it will be my last slide. Where our main objective is the deciphering of the human chemical exposome. So we understood that we have to merge all the initiatives at the national level. This is France Exposome. At the European level, this is IRIN. On larger, probably North America, on the southeast of Asia. And so there is a certain number of changes which appears. And the first one is the raw producibility across sites. We have to be able to compare the data, whatever the team was produced the data. And the second point is the consistency over time. And this is especially important when you want to interpret temporal trends, because temporal trends is a way of measuring the efficiency of the policies, and the reproducibility is once again a very important thing. Second point has been already treated, the high throughput capacity. For a medium-sized laboratory, we have probably possibilities up to 1,000 to 3,000 samples a year. The expectations of the community is above 100,000 samples a year. So something has to be changed. Robotic automatizations has to be introduced widely in laboratories. On the last point, my last comment, the control of the environmental contamination in laboratory, it is a real critical point. We are working on ubiquitous samples, ubiquitous hazard, These molecules are everywhere, starting from the gloves, from the solvent, from the consumables, and we need to move towards clean environment of work in order to decrease the pressure of these ubiquitous molecules onto the results of the analysis. And I think there is space for improvement. Thank you for attention.
Thank you so much. Michel, you have the last talk in this session.
Hello. I know you are a little bit tired first, and but first of all, I know now we are very familiar with all the exposome is and acronyms that you use and so on, and the technology, so it's very easier for me. But first of all, I would like to thank all participants to the half day on this symposium from INSERM, from University of Rennes and Masaryk, of course, but also the French School of Public Health, the French agency, ANSES, but also we know that Sébastien Denis was absent, but I want to thank Santé Publique France and the France University and all the members of the European Commission to be present here at Brussels. And I am very happy to be here because you know that it's a center of European democracy, and now we need that. So it's a very pleasure, Carl, to be here with you. And of course, I don't forget to thank David Alice and Gilles Bloch for the very kind words in the introduction of this meeting. So now it's a pleasure for me to present France Exposome. So I'm very sorry to be at the French level, because we come back at the French level, not at the European level, but it's very important for the France position. Okay, so what is France Exposomes? And you can discover the new logo that we create to explain what is France Exposome. So it's a new research infrastructure to support environmental health research, but also for the public health policy. And the main aim of the France exposome is to characterize a human chemical exposome. And it's very important that we focus only on chemical exposome for the human, and also to develop the interface necessary to link exposure and human health. So the main organization, the French organization that support France exposome is INSERM, and I would like to thank, of course, INSERM for the very implication in to create France exposome. But for other partner, it's Oneris that Bruno has already talked just before, and Université de Rennes, EHESP, University of Paris Cité. It's just a mistake in this slide, but I think the new name it's just a few days ago, and INERIS and also INRAE. So I move. So why you want to create a France exposome? Because there is a favorable French and European context. Of course, there is a positive dynamic with CPER. Carl talked in his introduction about CPER. It's a contrat de projet État-région, and it's very important to provide money to build new equipment. And also, I thank to PNS ECAT, so it's a fourth plan of environmental health from the French government, and it's a possibility to get money with PNS ECAT. There are also two pitch of two French ministers, who is very important for us. The Minister of Environmental Transition, Madame Barbara Pompili, but also the Minister of Health. So everybody knows Olivier Véran. So it was in the last May in 2021, and in these two speeches that promote research on exposome. So it's a very starting point for us. There are, as you explained this afternoon, many different projects in Europe, HBM4U, PARC, and we listened Pascal to explain what is PARC, but also we have a consortium. This consortium is based on the French structure that know very well each other, and we have a very long expertise in this field. So it's interesting to create France exposome on this base. And also we have a strong complementarity in expertise, but also a strong complementarity in a technological platform. And already mentioned, but it's very important to create based on the accredited laboratory, and it is the case also in health and is food. So I share the direction of France exposome with Bruno Lebizec, professor and director of Labirca. And the France exposome, it's a network of technological platform in the field of chemical exposome, with a laboratory of Nantes, laboratory of Rennes, but also the laboratory of Toxalim in Toulouse, and another one in the laboratory of INERIS at Verneuil-en-Alate, it's on the north of Paris, and also the lab of Robert Barouki in Paris Cité, University of Paris Cité. And one thing to know that it's at this moment all the partners are signing the memorandum of understanding. So it's a starting of this process, and it's a very important step. So the contribution of this different platform is mainly based on the human biomonitoring with targeted approaches, but also the suspected and the non-targeted strategy. And it's true for Rennes, for Nantes, and for Toulouse, but also on the toxicology kinetics studies, and also the modeling at Verneuil-en-Alate, and also predictive toxicology and system biology at the University of Paris-Cité in Robert Barouki lab. So if we want to summarize what is France Exposome, it's an academic network of platforms for the analysis of internal chemical exposure marker with targeted and non-targeted approaches. France Exposome, it's a part of the field in a different type of field, environment in food, but also in health. And we are, as a partner, à l'Envie, ANSES, Santé Publique France, but also INCA. We don't talk about the cancer research, but it's very important that INCA are involved in the France Exposome, and also Avison. So France Exposome will work as with already existing research infrastructure, and I can read, and it's mentioned in the left part of this slide. We need to talk with France Cohort, the Constance Cohort, France Génomique, and so on. It's already, the time is already finished? Wow.
5, so if we want to stay on time.
Okay, okay. So, well, the connection between France Exposome and other platform in France is very important, okay? And, okay, and, well, because it's a platform, we have many different equipment, and I have no time to explain how many equipment and describe all of them, but as already mentioned by Bruno, we have high resolution mass spectrometry, but also tandem mass spectrometry. And we have some of them already in the lab, and you can see in this picture. But a new one, and it's a very high technology, and what is the best actually in a commercial solution, and we can get a new spectrometry, mass spectrometry apparatus in a different lab. Well, and just to finish, I think, and I am very convinced that IRENE is a very nice home, and France Exposome will be the pieces of this puzzle, and we be sure that we have a very link to create with Recetox and France Exposome, and of course many other partners of IRENE. Okay, thank you very much.
Thank you, Michel. Thank you. It's very impressive what you've managed to do with the France Exposome. I don't think any Spanish minister would know the word exposome. I was going to say, I don't think any Spanish minister would know the word exposome, let alone use it in a speech. I think you've achieved a lot if you've managed to get it.
Well, yeah, but you know, it's a reason that I want to congratulate and thanks many support, because without this support, we're not able to do that. Yeah.
Right.
But if you want to say something,
I have a question.
Bruno, about France Exposome can be free, of course.
I have a question on the France Exposome. How do you see the place of epidemiology? I'm an epidemiologist, and I work with population studies, and your picture was mainly toxicology, obviously the high throughput platforms.
Well,
how do you link him with the? Obviously, you have a lot of epidemiologists in these.
In
places.
this slide, I can. No, it's. I cannot come back. But you know, it's a reason that I say that the link between France Exposome and the cohort is very important. Of course, first. And also, I lead a research institute of environmental health and occupational health, and we have many different epidemiology inside the IRCET. So, of course, it's very important. But here, it just focus on the process of the samples. So, you know, because you explain what is different European project on exposome. So there are many different biological samples generated, and but of course where the process are done. So at Recetox, of course, in US, but also in this platform.
I'm asking because I think one of the main steps that maybe we haven't made so much yet is the link in the within the exposome is linking the epidemiology and the toxicology, especially the functional toxicology, if you want to call it, the functional exposomics. So if we're looking at all the omics platforms in our epidemiological studies, we need to have test systems to validate, to replicate, etc. And we need to go both ways, obviously, with the.
Yeah, sure.
So this is, I think, in France you're in a great position to do this. Yeah, please.
Yeah. Well, if you like France Exposome, you will love France Cohort, which is the other, you know, France Cohort is the other, you know, infrastructure where all the cohorts are, you know, meet and it's, I mean, there's clearly a very strong collaboration between France Exposome and France Cohort, and there shouldn't be too much overlap. So that's, I can, you know, that's the reason why, of course, things are separate.
But it's impressive. Very good. So questions. We take a few minutes for all four speakers.
My question is about money. Most of the French scientists I work with, at least those I work with, and I mean also French research directors, tell me that EU research infrastructure projects are not that profitable. I mean, the money that they put on the equipments that they are used, and the resources, human resources they put into research infrastructures somehow are higher than what they actually receive. And of course, I'm aware of all the budget that has been given in during H2020 and Horizon Europe that has been cited during this presentation. But my question is, according to you and your experience, do you think that the funding that is given to the exposome community is enough for you? And if not, why? And the second question is how to encourage scientists from so many disciplines and communities to be willing to collaborate and contribute to the exposome community.
Yeah. So I can start the answer, and after Bruno can say more. For me, the most important, of course, you need a lot of money, so sure of that. I think the most important is to get new equipment and the apparatus, but also the human resources. And after, it's like facilities. When you have a project in France or from Europe, it's different because you can address your question, you can address your samples to this platform, to this facility. But the most important is that the facility has already the equipment ready to do something, and human resources. So I think the most important thing is there. And after we can, of course, it's not, you are not here to win money, because it's an academic project and it's very important to understand that. But where the state or the European community can help us, it's for these 2 points. Yeah.
Only a short comment. We have only 1 minute, but we are at the moment at drafting the business plan. So we know perfectly that the target is the equilibrium. Is not to make profits, obviously. But you can be sure that in 6 years we will reach the equilibrium according to our first evaluation. So this is very sure. And for the second question, it's always a question of communication, to be everywhere, every time, to say what we are doing and to invite the community to contact us and to contract us on coming back to the business plan and to contribute to the equilibrium.
We stop here?
I think it's time, but it was very, very interesting. Thank you, all of you, for this developments. But before the end of this event, we'll show you a short video of Monsieur Le Drian, which is Ministre de l'Europe et des Affaires étrangères du gouvernement français, that we can maybe see right now.
Mesdames et messieurs, au nom de la présidence française du Conseil de l'Union européenne, je tenais à vous remercier d'avoir pris part à ce symposium organisé par l'Université de Rennes et l'École des hautes études en santé publique en partenariat avec l'INSERM et l'ANSES. Ce symposium aura permis de clarifier le chemin que nous devons suivre en Européens pour conforter le rôle de notre continent dans la recherche mondiale sur l'exposome au carrefour des problématiques de santé et des problématiques environnementales. Jean Monnet considérait que l'Europe se fera dans les crises et qu'elle sera la somme des solutions apportées à ces crises. Et à bien des égards, la crise du Covid-19 a confirmé cette intuition. Elle a précipité la mise en place de l'Europe de la santé dont nous avons tant besoin pour protéger nos concitoyens et jouer tout notre rôle dans la construction d'un multilatéralisme sanitaire, de la solidarité et des résultats. Elle a accéléré les investissements nécessaires à l'Europe de la recherche. Elle nous a permis de prendre conscience que l'humanité ne saurait vivre en bonne santé sur une planète malade et donc de nous mobiliser pour jeter les bases d'une Europe des biens communs, conséquente et pragmatique. Mais les solutions aux grands défis du XXIe siècle, dont la pandémie nous a permis de prendre la mesure, sont évidemment des solutions qui doivent se construire dans la durée. C'est le sens de notre engagement en France, en Europe et à l'international pour donner à l'approche une seule santé des traductions concrètes et opérationnelles. D'une part, avec le panel d'experts de haut niveau Une seule santé, dont nous avons décidé la création aux côtés de l'Allemagne et qui a vu le jour en mai 2021 autour de l'Organisation mondiale de la santé, de l'Organisation des Nations unies pour l'alimentation et l'agriculture, de l'Organisation mondiale de la santé animale et des programmes des Nations unies pour l'environnement. Et ce nouvel outil nous permettra d'anticiper et de combattre les pandémies qui pourraient nous frapper demain. D'autre part, avec l'initiative internationale PRESOD que le président de la République a lancée lors du One Planet Summit de janvier 2021 pour mieux intégrer et renforcer les réseaux de chercheurs et d'acteurs en santé humaine, animale et environnementale qui, sur le terrain, luttent contre les risques d'émergence et de propagation des zoonoses. Là aussi, il s'agit d'un outil essentiel que nous appelons l'ensemble de ces États membres à rejoindre. Enfin, avec la réunion ministérielle Santé et Affaires étrangères que j'ai organisée à Lyon avec Olivier Véran le 9 février dernier dans le cadre de la présidence française de l'Union européenne pour renforcer la stratégie européenne en santé à l'international et traduire concrètement l'approche Une seule santé. Construire dans la durée cette Europe de la santé et des biens communs autour d'une approche intégrée des défis sanitaires et environnementaux, c'est aussi, je le sais, le sens de vos travaux. C'est pourquoi je tenais à vous assurer du soutien de la France tout au long de notre présidence du Conseil de l'Union européenne, mais aussi au-delà. Nous sommes à vos côtés et aux côtés de tous les chercheurs qui travaillent pour notre avenir. Merci.
Laurent Chambaud, in order to finish this event, a few words, maybe you don't have any. This is a microphone here.
Yes, a few words. Firstly, thank you everybody, all the people who attend this meeting or here in Brussels, and thanks Carl for these facilities, amazing facilities, but also all the contributors, and thanks also to the people who attend remotely, because I know that there were a lot. Also, thanks to all the contributors, and I think it's really a very important step for all of us, and it shows a really amazing and French and European scientific dynamics. And what also for me seems very important is that it's a dynamic on the exposome, and it was really amazing what has been said today, but on the broader sense, it's also a dynamic on what we can do on the environmental health issues, and with what we can do about the threats, but also the positive things. And I also noticed that many of you say that we have also to be very positive on this issue. The other thing I think is important to say is that I think that this initiative also shows a bridge between the scientists, between also all the institutional contributors, the academic institutions, but also a bridge with political leaders. And I think it's really important. And also, we don't have to forget all the society, because this also is an issue just to share with all the society, all that we have discussed today. I would like to finish perhaps with two very short things. The first one is that we will have different events after that. The first one will be in June in Rennes with the Atlantic Exposure Summer School, which will be in Rennes in the beginning of June. So if you want to be part of that, it's open. But also, I think it's really important because it shows a very tight link between the French presidency and the Czech presidency on this topic.
Mr. Dansack, next step, so...
Thank you. You said exactly what I want to say at the end. No. Thank you so much. I'm not expert in exposome, as you see, so I am from social sciences, and Professor Klanova, unfortunately, had to move to airport, yes. But she underlined sustainability. Talk about sustainability, and it's very easy for me because what I saw today. Thank you a lot, organizers. Thank you for inviting us and positioning Masaryk University among your friends and colleagues who can help with exposome topic. I feel it very responsibly, and I think that our experience is in disposition for other research units and universities. I see universities inside the problem which we bear on our in our minds, and have to do more with exposome. And as you mentioned, we will continue our effort next half of 22, as Czech Republic will continue in presidency within the European Union, and we plan to organize a similar event, not exactly the same, because our focus will be more on educational issues and outreach to public. And we plan it in fall period, probably October, November. Some issues moved our intentions aside, as Ukraine, So we are looking for a proper date for seminar on Exposome again. So thank you so much, and hope for for good results. We have a motto: don't start what you are not intended to finish. So. Thank you.
It's a good way to finish this event. Thank you to both of you. Thank you to all the participants. This is the end of this symposium. Have a good night. Have a good evening.